Join.
We can start, sir.
Okay. So good morning, everybody, and welcome to
Morning, sir.
the session of interaction and learning. Today we are going to cover three wide variety of topics across three lectures, and the first one around that is formulation and mixing technology. What I've done is I've kept the second presentation also available. So in case we save time, we will start the next one part of the next lecture also. The reason is that the last lecture of today's series is a long one, and our concepts which are new, and therefore we need to spend a little bit more time on it. So without any further ado, let me start on formulation and mixing technology. When we talk about formulation and mixing technology, what is formulation and sterile manufacturing process needs to be understood. Any injectable solution that are intended to be administered to a human body by intravenous, intramuscular, or subcutaneous injection are termed as formulation. So, formulation is nothing but an injectable solution which is given or administered to a human body. Of course, this when extended to other applications or other animals like veterinary products and all also it will still be called as a formulation only. Okay, the advantages of injectable include a fast onset and other parent routes of administration, reproducible, efficiency, efficacy profile, high bioavailability as a result of bypassing the oral absorption barrier, and suitability of administration under hospital setting. So, injectables products have a lot of advantages. First of all, because they are intravenous, intramuscular, or subcutaneous, there is a fast onset of the drug product getting into the body and acting. So the injections work much faster as compared to any other route of administration, be it oral or otherwise. It does not have to go through the oral absorption barrier, because when you take any oral medicine, it has to go through your reproduction system, sorry, through your digestive system, in which there are various kind of absorption barriers which occur, where it has to pass through the body into the intestine before it can kind of get absorbed into the bloodstream, and therefore it has a longer time before which the drug is available. It is reproducible. I mean, so are other formulations, of course. Has high bioavailability, meaning that it is the entire formulation can be directly be available into the bloodstream, rather than having other components which have to overcome the absorption barriers which are there in the other routes. And it is suitable to administer under a hospital setting, whereas in case of oral absorption, sometimes you have to leave it to the patient to take a medicine, and they may or may not take it. Here, because it's an injection, you need a nurse to administer it, unless they are of course the self administration injections like insulin and others. In order to meet the safety, efficacy, and quality standards of parenteral dosage forms, injectable solutions have to be sterile, low pyrogen, and meet the requirements of Compedia specification, such as greater than 90% label claim, related substances lower level, then the tox qualified level, content uniformity, pH, osmomolality, particulate matter, essentially free of visual foreign material, etc. So while we talk about injectables, it is very important to understand that these solutions, while they may have its advantage of a fast onset and route of administration, need to be free from any pyrogens, have to be sterile for sure, and they have to be available meeting the Compedia specification, meaning that you have the US pharmacopoeia, you have the Indian pharmacopoeia, you have the Chinese pharmacopoeia, you have the Japanese pharmacopoeia, you have the European pharmacopoeia. So it has to meet the specifications from a perspective of those pharmacopoeias for a given drug product. And the availability of the drug has to be greater than the 90%. So if we say it is going to be 500 mg of a particular drug product, then greater than 90% of that 500 mg has to be available in a given product to be qualified. In fact, it is now that it has to be 100% or more than 100%, so that there is never a situation of a lower drug dosage given to a body. Of course, it has to be uniform, it has to be at the right pH. Why? Because it is getting into the bloodstream directly, so there is no pH adjustment possible. When you take an oral drug, it goes into your body, it goes through the digestive system where there is a breakdown, and the final drug product, when it gets absorbed into the bloodstream, there is a certain amount of pH stabilization which happens. Here, since you are directly administering it intravenously, the pH has to be right, because if the pH is too high or too low, then obviously it could, while it could have an effect on the formulation, it could also affect the patient. uh, of course, it has to be free from any contamination, be it particulate matter, foreign matter, etc. The first step in injectable manufacturing is obviously the formulation development. It involves extensive research and development to create a stable and effective drug product. So it is important to understand how formulations are developed, and we are going to cover a part of this in this particular session. Formulation scientists work to identify the appropriate active pharmaceutical ingredient, excipients, and delivery systems for the injectable, meaning that they have to see, okay, what is the drug substance which I'm looking at. Active pharmaceutical ingredients would hereafter be referred to as drug substances, because they could come from a biomanufacturing process, or they could come from a chemical synthesis process, or a combination of a biopharma and chemical synthesis process. It has to have a certain amount of buffering agents to maintain the pH and osmolality, which is given in form of the excipients here, and then the delivery system, meaning whether you are going to put it in a vial and therefore somebody will administer it. So is it a multi-dose? Is it a single dose? Does it is it a cartridge? What is the quantum that one has to take, whether it is 1 ml, 2 ml, 3 ml, 5 ml, zero point 0.5 ml, etc. Whether it's better to formulate it and fill it in a pre-filled syringe or an ampule or a vial. This is something that a formulation scientist works on. As such, this topic is still growing. There are various developments which are happening because of the advent of biopharmaceuticals. Aseptic process is becoming mainstream patient care now. Also, there are efforts which are going on in terms of formulations and in terms of drug development, where when a patient needed to take multiple doses, for example, anybody suffering from diabetes normally needs to take, and if they are on an insulin injection, they generally need to take an injection every day, sometimes multiple times in a day. And that is a very painful process, because if you have to poke yourself with a needle every day, obviously it's going to be a very, very painful process. For cancer treatment also, there are some treatments available where the patient is required to take injections almost on a daily basis, or sometimes multiple doses in a day, and that becomes painful for the patient, apart from the pain they are already going through because of the condition of the disease. So as such, formulations are being worked out where you take the injectable, you push it inside your body, and you do a sustained release. So it's a long acting injectable where the product is formulated in such a way that it goes into your body and stays there, and it releases a very small amount of micro dosages of the drug substance into your body through the bloodstream in a manner that is required for a day. So a cancer patient, for example, need not take an injection every day. They may be able to take an injection once a week, and therefore their pain goes down, because now they have been injected by the drug, but that drug is inside their body for the next 1 week and is releasing in the right quantum. And this is definitely a lot of science going there, because the formulation scientists have worked out the mechanism around it, saying how that active ingredient will go out in the right dosages on a daily basis. Okay. So these are developments which are happening. So this is a field where there is a new thing or a new formulation, new way of delivering a drug which is coming into the market, and it's very interesting to be studying around how these formulations get developed. It also poses challenges in the manufacturing processes, because you are used to a standard way of manufacturing a process, and when it comes to these kind of products, there is a different methodology of manufacturing the product itself. Of course, the last step of formulating it, putting it into a vial or lyophilizing it remains the same, but nevertheless the preparation stage to reaching that while filling is a different process altogether. By 2030, it is projected that the global biopharmaceutical market will reach a revenue of $856 billion, and is predicted to expand at a compound annual growth rate of 12.5%. So 2030 it will reach 856 and then continue to grow at 12.5% year on year. Covering a very broad range of treatments, biopharmaceuticals are most often administered via parenteral injectables. Most of the biopharmaceutical products that we looked at yesterday, whether it was insulin, whether it was the monoclonal antibodies, have to be administered through the injectable route only. And therefore, this market is going to also reach about 150 billion. The parenteral injectable of biopharmaceutical, just the formulation part of it, is going to reach $150.65 billion by 2028. There are many reasons behind the growth in this market, with the key driver being the billion of messenger mRNA inoculation that continue to be delivered globally. So we saw yesterday how a bio manufacturing process works, and we found that RNA, the DNA, the RNA, and then the protein coming out of that are an important aspect. Even when we look at COVID-19 vaccine, we know that one of our vaccines developed in India was developed through the mRNA route. And this is now becoming a more and more researched program, where you see that a lot of drug formulations are coming through the biopharmaceutical route and through the mRNA route, be it vaccines or be it therapeutic medicine for treating a disease. So formulation is a very, very important aspect of the sterile manufacturing process. What are the challenges which are there in the formulation dosage or a parenteral dosage form? Is one is the formulation should be stable. It should be stable. It should not disintegrate. It should not kind of change its physical chemical properties, etc. So that's the most important part. The compatibility of the drug substance with the packaging component. So you will have fill it in a vial which is made out of glass. You may fill it into a plastic container. You may fill it into a PFS. So it is important that the drug substance is compatible with those packaging components. There has to be a sufficient drug concentration with a reasonable pH range, and without using excipient levels that cause blood incompatibility and tissue irritation issues. So this is a very important statement, because what we are saying is that we need to have sufficient quantity of drug available in the formulation that we are doing, and it has to be at a pH range which the body can accept, because the moment you inject something into a body and it finds that it is not compatible or starts to cause an irritation to the tissues within the body, obviously the drug product will not be effective, and the person or a patient may react to injection, which is something that has to be avoided. So it is very important that we do a comprehensive characterization of a drug. Now, let us understand what is involved in a parental formulation development. It is consists of these steps which are listed down there. So there is pre-formulation studies, then there is prototype formulation development, then there is accelerated stability studies, packaging selection, process development to evaluate critical process parameters, scale up of the formulation itself, manufacturing of demonstration and GMP batches, long-term stability studies for up to 36 months, 24 to 36 months from a physico-chemical stability perspective. And apart from this, we also need to evaluate the stringency and the control requirements for the assay of the drug product, the leachable, extractable, sterility, pyrogen issues, particulate matter specifications, etc. Now, in such a situation, what happens is that as you can see, the study or the development of the formulation needs to follow some of these steps. Obviously, some of these steps are happening in parallel. Like for example, while the pre-formulation studies are going on, packaging selection can start happening, because you need to worry about the compatibility of the drug substance. So you will start selecting the kind of package you would do after the pre-formulation. Only you can do the prototype formulation development, and once that is done, then the accelerated stability studies can start. But at the same time, while prototypes formulation development is happening, you can start to define the critical process parameters, and therefore do the process development work to a certain extent during that stage, so that when you are ready with the prototype formulation development, you are in a stage where you can quickly turn around the process development. When you're doing the process development, you start looking at scale up parameters and start to scale it up. Only after you've done all this, and unless you have a good manufacturing practice or a batch which is produced under aseptic conditions or under sterile manufacturing process, if it was a terminally sterilized product, you have to manufacture these batches in a certified facility, and that batch what you get out from such a facility can be used for clinical studies, can be used for long-term stability, etc. You cannot just use any lab formulation for clinical studies. Of course, you can do some preclinical studies with the formulation developed in the lab, as long as there is no animal or patient involved in the study. Okay, so these are the various stages. We're just going to quickly go through some of these stages. I have a lot of data which is available, which is there for you to read and understand this, but let's understand the critical activities which go at each of these stage. During a pre-formulation studies, what are the parameters, or what are the target deliverables from a pre-formulation study? First is to ensure that there is solubility and stability as a function of pH. So if we are going to prepare an injectable at a particular pH, which is generally a neutral pH, maybe towards slightly towards 6 to 8 or around 7, then we have to make sure that the formulation is stable, that the drug substance going into the along with the excipients or the buffering agent is stable at that pH and does not lose its properties, or lose its efficiency, or lose its acid, does not degrade it, does not start to create other impurities when in such a situation. So solubility and stability are two major this thing. We determine what is called as an ionization constant of a dissociation constant. So what this means is that if the product were to dissociate, it would form an ion, it would form ions, and those ions become reactive. So we need to determine the dissociation constant and the ionization constant, saying that when I'm making this formulation, what will be the extent of dissociation and ionization which can happen for this, and how do I prevent it, because we don't want this to happen. Any partitioning, in the sense any separation of the different components, whether sometimes when you have let's say suspensions, the products, the solid product tends to settle down. You may have an emulsion where you may have an oil or a polymer with water. In that situation, you should not see a separation of those layers. It should be a uniform mixture, because when you are doing it, of course, in suspension, if it the settles down, you have an option to shake the bottle or the shake the vial and then administer it. So suspensions, and today we are looking at nano suspensions where the settlement happens like once in if you leave a solution for 5 days only, then you see a clear partitioning or the settlement happen. Otherwise, you can't visibly see it also. But nevertheless, any suspension which is there before it is administered has to be properly made uniform by shaking it before it is administered. Moisture sorption and desorption, very important, because if you have a drug product which is going to start taking moisture, you could have effects of either the drug substance being too dilute or the drug substance being too concentrated. Polymorphism and crystallinity by different DAC, thermography analysis, and powder X-ray diffraction. So this is the property of the powder. When it is solubilized, you have to look at it through these calorimeters and TGA and X-ray diffraction. Salt form screening. Does it form any kind of a salt when you add it to the buffer? Does it form a third column quantity salt? Sometimes it may precipitate out as a salt, or it may form a salt which just stays in the solution itself. The stability of the drug substance because of these various parameters of the formulation. Assessing the compatibility of the API and excipients using stress testing and force degradation to evaluate the effect of pH, temperature, humidity, light, and oxidizing agent. So you now make a formulation and you subject it to testing. You force it to degrade. You put it at a different pH condition and see what is the effect. You put it at different temperatures for a given pH and see what is the effect. You change the humidity around the formulation at different levels and see what is the effect. You expose it to light. Light is the natural light. You expose it to monochromatic light. So a lot of drug substance or formulations are actually not stable to when you expose them to the natural visible white light. So when they are manufactured, they are manufactured under monochromatic light conditions. So you use light wavelength of 590 nanometers or more to kind of when you are making the formulation. So this is an important feedback to somebody who's designing the facility, because if a formulation is not stable at natural light, you need to make provisions in that facility to provide monochromatic lights or specific light conditions so that the product does not degrade. There are a lot of products which are in this category. In addition, compound solubility using commonly used solvents, co-solvents, surfactants, solubilizers, and complex agents will be useful in setting the direction of formulation development. So it is very important to see whether the compound is soluble in the commonly used solvent. Water is the most commonly used solvent, but there could be other co-solvents where you use a slight amount of buffering agent, or you use a surfactant or a stabilizer or a solubilizer. These are certain salts which you add, or certain compounds or molecules which you add, so that the formulation is stable. So the compound has to be soluble and has to remain stable. So this is what is involved in a pre-formulation study. Of course, there is the degree of ionization that we talked about, and I'm not going to cover it in this introduction session of formulation. But those who are interested can deep dive into this. There is a Henderson-Hasselbalch equation which is used for arriving at the pK values. Okay, the effect of the pH on API solubility and stability is most important, and there is a graphical representation which we have which are taken from one of the literatures, where if you look at the pH, the pH was around 3, for example, and as you drop the pH, what happened was the solubility also started to drop. So the pH was at 0, and there was a solubility at 5,000 micrograms per milliliter. As the pH started to rise, you could see a rapid degradation of the solubility. At some point, it just kind of went to 0. So if the product is if the compound is not stable at a particular pH and or is not soluble, then it becomes a problem. Now you have what is called as a pH target. You have a pH target because you are seeing the rate constant at which it is dissociation and ionization are almost kind of at a stable range here, and the pH is also kind of remaining stable here. So you would probably take a pH target here and say, okay, I will keep this solution at a pH between 2 to 3 so that I get solubility of around 2000 micrograms per meter cube, or between 1000 to 2000, and I can use the solution at this dosage rates. Now, whether can I inject this into a body or not, it would be a study which would be done, and how the body reacts would also be there. It is some, and then you add also a degradation compound to check this. So you will degrade it. So you did degradation studies or stress studies, and that is how you arrive at the pH constant. So this is the important aspect of solubility and stability at a given pH, which is important. Salt or co-crystal formation is another factor which we need to look at, but that we have already covered, so I'm not going to get into it now. What is a prototype formulation development? It covers the selection of the buffer. What will be the buffer? Now you've arrived at what pH I want to make the solution. I know what is the solubility range. I know what is the constant, dissociation constant. Now, based on this, I have to do a prototype development of the formulation, where I select the buffer, I select the stabilizers, the any isotonicity agents, any antioxidants or preservatives that I need to add to keep the compound stable, and solubility and viscosity enhancing agents, because you need to make sure that the product remains stable. Most of the injectable products have a shelf life of 2 years, are expected to have a shelf life of 2 years, and in such a situation you need to make sure that during the entire shelf life it remains at a uniform condition of physicochemical properties. Okay. pH and buffer selection, since pH is most important criteria, and you have a pH which can be acceptable from 2 to 11 for intravenous injections and intramuscular injection, and 4 to 9 for subcutaneous injections. Because of potential irritation issues, you need to choose the right buffer. Now, there are various buffers which are used. There could be tris buffer, there is the acetate, phosphate, citrate, and other buffers. These buffers are generally in the range of 5 to 8. So, as I said, trying to keep the solution more towards the neutral range of around 6 to 7 is what is generally preferred, depending on the solubility of the compound, but is something that we need to look at. So the right buffer is selected during the prototype formulation development. So you will use different kind of buffers like acetate, phosphate, tris maybe, and then formulate the compound to see whether it is remaining stable at this stage. The buffer capacity should be kept to a minimum to enable body fluid to adjust to the formulation pH quickly, or to the physiological pH. So the body itself has a physiological pH, and if you try to keep the buffer at a pH which is way different from the physiological pH, then obviously the body needs to adjust before it accepts the drug into the body. For that reason, the buffer should be kept to the minimalistic and should be preferably kept as close to the physiological pH as possible. Okay, otherwise irritations may get caused if the pH is too much variance between the physiological pH and the formulation pH. Then you could also have issues about salt formations when the pH changes, so you don't want such precipitations occurring inside the body. Now you have to visualize this from a perspective that when somebody is injecting something into your bloodstream, the blood is like a flow path. It's like a continuous flow path being pumped. It's like a pipe having a lot of circulation. The heart is a pump which is pumping the blood continuously into our body from top to bottom. And in that, you are now introducing injectable product. When it is moving through the entire body, it should not cause irritation. If it should not have a microsite where it goes and forms a salt, because if it does form a salt, it will start to create an irritation into the body. So, given this, buffer selection and pH is what you have targeted a pH, and to achieve that, what buffers you will use, which one gives you the maximum solubility as well as the stability, is something that you would do in the prototype formulation studies. Osmolarity, they should be ideally isotonic wherever possible. So you use dextrose, mannitol, glycerol, and sodium chloride as the most commonly used agents to adjust the tonicity of the solution. What happens is that it is important to make sure that when it goes inside the body, again, you don't have again any issues of the osmosis happening, and therefore again a salt precipitation which is happening. So from that perspective, osmolarity is important. You use dextrose, mannitol, glycerol, and sodium chloride as the most commonly used agents. Why? These are also the agents which are used when a patient, when a person has a problem of, let's say, weakness or something. You say, it's a common this thing, glucose chada diya, right? That glucose is nothing but actually a dextrose or a mannitol solution, or sometimes it's a mixture of sodium chloride and dextrose, which is given to a person to provide the stability of the bloodstream to and create isotonic to maintain the isotonicity inside the body. So from that perspective, these are the most commonly used compounds in the formulation to make the solution isotonic. Solubilizers, sorry. Due to a high percentage of insoluble compounds in the development stage, your API may be insoluble at a certain with the water itself, and you need to solubilize it. You need to use certain solubilizers so that you can make the drug substance solubilize in the product. So again, there are various kind of compounds used like propylene glycol, polyethylene glycol. Very, very important to understand that you have to use these very, very carefully. One of the recent incidents of the Ghana cough syrup was because it was requiring the use of a glycol, but instead of using a polyethylene glycol PEG, the company used monoethylene glycol, which was wrongly used, as a result of which those deaths occurred. So making sure that the right agent is used is very, very important, because it can create havoc otherwise in the formulation itself. Okay. Container enclosure, very important. You use different type of containers which may be made out of plastic, which may be Teflon coated, which may be siliconized. And therefore it is very important that when you pour the injectable solution into it, there should be no interaction. There should be no extractable or leachable coming out of the container into your drug formulation product. Extractable leachables we have covered during the single-use topic, in which it was very important that any product which you fill in a bag, you should ensure that there are no extractables or leachables. There will be extractables and leachables, but they should be within an acceptable limit that it does not cause a harm to a patient. So in that scenario, choosing the right container and closure becomes important. Most of your pre-filled syringes and cartridges are siliconized. They have a thin layer which cannot be seen by even the eye of what is a thin silicon layer, and your drug product is actually touching that silicon layer. Now that silicon starts to leach out and get into the formulation, then that silicon is going to go and sit inside your body. While the body can accept a certain amount of silicon inside itself, or some of these polymers like Teflon, etc., it cannot be to an extent which it starts to create a detrimental effect. So very important to choose the right kind of a container and closure, because the definition of a container or a closure comes from the fact that it is based on the method of by which you are going to administer it. If it was a large volume parenteral or an infusion product, when somebody is admitted to a hospital and they have to be administered drugs, you see generally those parenteral bags which contain saline solution or dextrose solution, which is hanging. The nurse will come and put one injection into that bag. Okay. Today, of course, things have changed. There is a advent of formulation there also. So instead of allowing the nurse to inject something, the formulation inside the bag comes pre-defined, meaning that you could, if you needed to administer, say, paracetamol to a patient who's admitted because, with a complaint of high fever, then that bag itself contains the paracetamol with the necessary saline solution or dextrose or whatever carrier solution which is there, so that the nurse does not have to come and keep on injecting. Why was this done? So that there is no maltreatment or malpractices which are followed. Of course, most of us know that sometimes, the nurses can forget to dose that thing, or may have an issue of overdosing or underdosing if they don't follow the processes right. If there are say hundreds of patients there and the nurses running around, obviously there are mistakes which happen, because they are at the end of the day human beings. To avoid such situations, there are pre-formulated injection bags which are now available. So for all these purposes, it is very important to understand that the container and closure are important from a perspective that it should not convert the injectable product into something which will be detrimental to the patient. So these studies are also done alongside with the prototype formulation, can be started from a pre-formulation stage. Process development and scale up. Now you have developed the process. I'm going to use tris buffer. I need to use X quantity of tris buffer to go with the drug substance dissolved in water and make the pH 5 to 8. I need to do the dissolution under monochromatic light. I need to make sure that it is dissolved at a temperature of, let's say, 25 degrees centigrade, and then I add the drug. First, I make the tris buffer, then I make the drug substance by dissolving it into another container which has got water, which has got solubilizers, etc. And now I mix the two and mix them pretty well, and then I fill it into the container, the final container, which could be a bag, which could be a vial, which could be a pre-filled syringe, which could be a ampule. So from that perspective, you have established all these parameters in the process development or the product development study. Now you have to carry out the larger process. You need to make sure this process is sterile. You are not introducing contamination. So how do you make this entire process which is free from any pyrogens, free from any contaminants, free, is stable, no particulate matter, it can be filled in class 100 areas, all that. So all that we studied on sterilization, we said different type of sterilization techniques, etc. How do you make this product sterile? Can I do it by filtration? Do I need to do terminal sterilization? That is established. Now that is established, how do I need to actually carry out this process? What do I do first? Do I take water first, or do I put the buffer first, or do I put the, let's say, polyethylene glycol first, then I put the drug substance into it? Because the sequencing of how you mix different components, how you put the ingredients inside a formulation also becomes important. Just like when we are doing any kind of like say dish in our homes, our mothers cook in a certain sequence. They do first, they put first a certain ingredient, then they put another ingredient, then the third ingredient, then they mix. They don't start with putting the last ingredient first and then put the other ingredients, because they know it won't cook well. Similarly, when you are doing a formulation, it is very important to have the sequence of it right. Also, to ensure that, to if you are targeting a sterile product, do you need to filter the water before it is taken? After you made a buffer, the buffer could be made by taking water and adding a buffering agent or a salt. In that situation, do I need to mix it, and after it is a uniform solution, do I need to filter it before I put an API into it? What kind of filter do I use? Okay, so all this is a study which you will do during process development, and you will also worry about what are the parameters, at what agitation speed do I get the mixing, what are my mixing parameters, do I need to do a size reduction, do I need to, am I having an emulsion kind of a situation which I need to homogenize? So from that perspective, the entire process development and scale up parameters are established. It also allows us to select the type of tubes, type of filters. So whether the filter should be PVDF, whether the filter should be PTFE, whether the filter should be hydrophobic, hydrophilic, all these decisions have to be taken at the time of process development and scale up. Because when you're doing it in a lab, you are not worried so much about the sterility, though you will try and do it as cleanly as possible. You are still not into a microbiology environment. You will get into a microbiology environment only when you either developing that product in the lab for clinical studies, or you are doing a process development. So in that scenario, everything that goes into the scale up, into the large final process, becomes very important. Okay. Compatibility with infusion sets. I spoke about the infusion bags, plasma and blood compatibility. So this is very important, because compounds which are administered intravenously must be compatible with the plasma and blood. So there is blood and plasma tests which are done, where there should be no precipitation either of the compound or either about of the plasma itself, because it can create conditions of clumping or hemolysis, and can create even adverse reaction, which could have a human being either having defects because of these formulations, or can also result in a death of a patient. So during process development and scale up studies, these tests are done to make sure that you have the formulation right. It should not get into issues. Then, of course, stability. You have to have a stability which is minimum of 18 months. That's what the demand is from the pharmacopoeias and regulations, that should the minimum storage, and it should be preferably at room temperature, because you cannot afford to keep refrigerated solutions when it is a product which is being dispensed, because at the end of the day, a pharmacy is giving it to you, or a hospital is giving to you. If all the products happen to be requiring refrigerated storage, it becomes difficult. So it is important that the stability is checked, preferably at room temperature. However, it is very likely that a lot of biopharmaceutical products, a lot of vaccines cannot be stored at room temperature because they are not stable, their shelf life is down. And since we need this minimum storage shelf life of 24 months, ideally, we would not like to degrade the product, and if refrigeration is the only solution to it, so be it. So if you all have noticed, when COVID-19 second dose was being given, the hospitals used to bring in the wires into a cold case, and they used to leave it in the cold case and gather the number of patients so that the entire vial, once opened, they can keep on administering. So it was a 5 ml vial, and 0.5 ml was what was being given to the patients. So they needed a minimum of 10 people to be available so that they open one vial, take it out, and start jabbing it. Why? Because they were stored at 2 to 8 degrees centigrade in a nice controlled temperature controlled area. They took it out of it and put it in that ice box, and that ice box is not going to stay to 8 for a very long time. So they needed to make sure that they had sufficient number of patients, so that the moment they took out this vial from the main storage and started to administer it in the shortest possible time, it was administered to the patient, so that it did not change its temperature condition, and therefore its stability did not get affected. It was not that the vaccine was so unstable that it would degrade immediately, but it is a care that the nurses and the doctors take, so that every patient who's administered is getting the right quality of the drug. If you leave a vial open for a long time and the temperature goes to, let's say, plus 12 plus 15 degrees centigrade, obviously the patient who has taken the vaccine when it was at plus 15 degrees centigrade versus a patient who took it when it was 2 to 8 degrees centigrade, there is a certain change in that physico-chemical property of it. So therefore, from an administration perspective, you haven't administered the same kind of compound. So these are some of the nuances which are very important. So the storage stability studies are done. How are stability studies done? The product, when it is formulated, when the process is developed, produced in a GMP facility, batches are taken. Those batches are kept in large walk-in incubators, which are at different conditions. There is ICH, which is the International Harmonization Committee, which has defined certain conditions at which the product should be subjected to stability. And apart from cold storage, there are four other conditions of temperature and humidity at which these products are stored. What you do is you store, let's say, 20,000 vials, 30,000 vials, or cartridges, or PFS, or ampules into that chamber, and you take out one vial or a couple of vials and check the physico-chemical properties, even the microbiology properties, at a certain interval. So you will take a sample, let's say 1 week after you have produced the batch, check it out. Then you will take it after 2 weeks. You will take it after 1 month. Then 2 months, 3 months, 4 months, 5 months, till 18 months, sometimes to 24 months to 36 months. You continue your stability study program so that you can establish how long is that product stable in a given condition. Why are these conditions necessary? Because when you transport these, the manufacturing happens at one site. From there, it is transferred to another distributor who may have a go down somewhere, and sometimes you even ship it across countries. Now, when you are shipping a product from, let's say, one country to the other, you are either using a ship which goes through the sea route, or you may be using a flight which may be taking it out. Now, you may have stored it at, say, minus 20 degrees centigrade at your facility. Now you dispatched it, the flight person took it, he took it on a flight, and it landed in, let's say, Charles de Gaulle airport at Paris. And in Paris, the minus 20 degrees centigrade refrigeration area was not available for some reason. Then, but 2 to 8 was available. So what the airport guys did was, or the guys said is, okay, put it at least in 2 to 8 degrees centigrade. and within 1 or 2 days we'll create the space. So it went through a variation of conditions which is there. Now you shipped it from India, which was at a different temperature and humidity conditions as compared to what it is in Paris, what it is in United States of America. So in such a situation, when you create the stability studies, you are establishing parameters which are different conditions so that you are 100% sure that your product is going to stay stable for those 2 years and it can be administered to a patient. Most of your drug substance, including your formulations, including your medications which you buy from a pharmacy, have an expiry date. And if you look at it very carefully, generally between the manufacturing date and the expiry date, there'll be at least a 2 year differential. Sometimes more than 2 years, because the stability studies have proved that it could be stable for longer, and therefore they have submitted the necessary papers to the regulations, and regulations have approved that yes, you could put that date on the thing that this product can be used for up to 3 years, for example. In certain cases, you see 2 years, certain cases, you see a lower value. So when you buy a medicine, also always check the manufacturing date to the expiry date. Do not buy expired products because of the reasons of the fact that you are now more educated and more knowledgeable, saying why I should not buy an expired medicine. Second is that if something is very close to expiry and your course is for 3 months, you go to a pharmacy and you are supposed to buy a medication. That medication was produced somewhere in 2022, say December, and you are taking this drug today, and the amount of medication which is prescribed to you can last you for, let's say, 6 months. Then you are actually taking the drug at a very final stages of its shelf life. So these are important aspects to look at and to make sure that we are conscious about not buying expired medicines or near expiry medicines if the course was long. As long as it is uh, consumed within the expiry period, it is still okay. But if it is a longer duration medication which is prescribed, then it should not be taken. So stability is an important aspect in formulation to determine what that storage is and what condition it is. Okay, so with this, we come to the end of formulation as a topic. I have tried to cover the key aspects and not gone into the depth because it was only an introduction which was required. Now, let's look at mixing. Mixing as a technology is quite evolved today. Mixing is very important because when you are doing any formulation development and here you're dealing with liquids, you need to make sure that the mixture is uniform. It is, in essence, mixing all the ingredients which you are putting into it. It does not lead to any contamination because you are looking at trying to do the formulation in a sterile environment or at least at a low bioburden. Okay, and if you were doing an aseptic process, then obviously the mixer has to make sure that it has to also meet the aseptic requirement. Now, why is this important? I'm going to go 2 slides up and show you some arrangements which are there. Now, there is always going to be a drive or a motor which is going to impart the power to your agitator, right? Because it has to rotate. You provide electrical power, it may generate a magnetic field or it may be a direct coupled. There will be a motor will have a shaft. That shaft will get coupled to a agitator and the agitator will rotate. Simple arrangement. Now this motor is made out of mild steel, cast steel. It cannot be inside the process environment. You cannot sterilize this, you cannot sanitize this. There is a coupling there and it has to go through your tank. It has to go through your tank body before the agitator is installed. So there is a opening in your tank through which your agitator is passing, and therefore that opening needs to be closed such that it does not allow any contamination to come from outside to inside, and also does not allow a leakage from inside to outside. So that sealing becomes very, very important, and that is why the emphasis on the fact that you need to have a mixer which prevents contamination and can be mixed using aseptic techniques. Okay. Depending on the type of formulation being manufactured, there is a need to use different type of mixtures. So you need to decide what kind of an agitator I need, because it depends on the formulation that you have developed. If the formulation requires low shear, does it need an emulsification, is there too much of powder solubilization which is involved? Depending on the application which is there, you have to define the mixer, the type of mixer which is used. Sometimes in a multi-product facility, you might be doing multiple of these actions. So you may have a low shear application, you may also be doing powder solubilization using the same set of equipment. So you have to use something which is going to meet both those criteria. Okay, so that is very important. Ensuring that the mixers are constructed to maintain sterility and can be in situ sterilized are two key requirements while ensuring uniform mixing at the lowest possible power and time. Why is this statement important? One, that it has to be able to in situ sterilizable. You are going to pass steam and sterilize the vessel. If you cannot sterilize the mixer which is there, then you will fail your sterility assurance criteria. So a tank comprises of lot of components, nozzles, etc., and agitator is one of the key parts there which needs to be sterile. So if you look at very large fermenters, we spoke about biomanufacturing. They need agitators. If you talk about a 60 kL or a 100 kL fermenter, the motor is going to be something like a 200 HP motor, and the shaft size is going to be like 155 mm. Now, when that shaft is going into your vessel, obviously there is a big opening there, 6 inches opening. You need to close that opening. So you use what are called as mechanical seals. But these mechanical seals are also sterilizable. So you sterilize the seal and you sterilize the inside of the vessel so that you ensure that nothing from outside to inside can go, or no contamination can survive in that environment. Why is this important? Because while most of your formulations and injectable will last no longer than a day, or your processing time is not longer than a day, in case of fermentation, you could have cycles of fermentation which go for 7 days, 14 days, etc. Now, in such a situation, the agitator is continuously running, going round and round and round and round and round. And during those time, if there is any breach of that seal, as a result of which a contaminant gets inside your vessel from there, then your entire batch goes for a toss. You have to discard that batch. So in such a situation, you need to make sure that the mixer design is very well studied, and it will maintain your sterility. It will be in situ sterilized. In case of formulations in the injectable, we use magnetic mixers. We do not use mixers where there is a direct insertion of the shaft all the way from the motor into the vessel. So what is generally done is that there is a weldable tank plate. So there is a magnet here, and there is a stator here. So this is a static magnet. This is a rotating magnet. This rotating magnet goes inside this cup here. This is like a cup. This is like a tumbler, ulta tumbler. You put the magnet inside it and rotate the magnet fast. When you rotate the magnet, what does it create? It creates a magnetic field. That magnetic field in turn creates a magnetic field around this static part, and this mixing head, as it is called, or an impeller as it is called, will be sitting on top of this post, and it has a magnetic field. So magnetic field, when it is generated, it causes a deflection, and as a result of which the impeller starts to rotate. Because the motor is rotating at a certain speed, and it's creating a magnetic field, the magnetic field in turn is creating the impeller to rotate. So this is the technology which is used today in most of the formulation vessels, as far as injectable is concerned. But the reason I brought the concept of having mechanical seals was because you were also talked about biomanufacturing, and in biomanufacturing, you cannot use magnetic mixers all the time. You may be able to use magnetic mixers in certain applications, but not in all the applications can you use magnetic mixers. In such a situation, you need to use a metallic shaft which is coupled to a motor, goes into the vessel, and therefore needs to be sealed at the opening on the vessel itself. Now, there are what are the applications and what are the different type of agitators which are used. There is a low shear application. So most of our biopharmaceutical compounds where you have proteins, which you may have cells, for example, and final fill solutions may be sensitive to shear. If there is too much of a shear onto it, it will start to disintegrate. The proteins will start to break or the cells will start to disintegrate. In such a situation, you need to make sure that the mixing is sufficient to create a homogeneity to allow you to have a proper mass transfer and heat transfer, but at the same time it is gentle on the product so that it does not damage the product. Okay. The shear sensitivity, as you purify the product, the shear sensitivity also goes up. Protein chains that are exposed to too much of shear will break and form agglomerates, which can be indicated through cloak filter or failing product quality. And therefore, it is very important to choose the right kind of mixing system for your low shear application. Now, there is another part which is emulsification. Some of your formulation, as I said, are oil water mixtures or polymer water mixtures, and therefore you need to have a very uniform emulsion which is formed. How do you, when you mix oil and water, if you mix it with a spoon in your house and leave it, what will you see? You will see some fish eyes or lumps. You will see some oil globules which will be floating very haphazardly within the mixture, and you will see the water layers, and then you will see a oil layer and separate at the top. You don't want these lumps to be there because the drug substance, sorry, the API is actually encapsulated in either the oil or the water globule. And if that is non-uniform, then when it is administered into a patient, then he won't get a uniform drug into the body. So it is very important that for emulsifications, you do a right kind of mixing. So how are those mixings done? They are done by using high shear mixtures, which are a rotor stator design. So I'm going to show you that. So it generally looks like this. There is a tank plate, there is a rotor. So this is a rotating part and this is a static part. And if you look at this, this has got something like a teeth that you can see here. So when the liquid goes into it, it gets crushed, it gets kind of mixed at a very fine level. It is like when you create when you have a very high speed mixing which is happening. That is what happens between a rotor and a stator. So you are crushing it, grinding it. So particle reduction happens in one way, and it homogenizes the oil and water globules. It breaks it down so much that the oil and water tend to form a uniform mixture. If you want to do an experiment, take oil, take water. You have the handheld blender in your house, right? Just put that into the water and oil and run it for maybe about 2-3 minutes, and then you remove it and you see the mixture. You will see an emulsion form there. And that is what a rotor stator does, that it puts a large amount of energy at a high speed and crushes the particles to such a small size that they get to with each other and form a uniform mixture. So during emulsification and high shear applications, you need to use those kind of mixtures. Powder and vigorous applications, the biggest challenge is that when you're working with powder, the powders could have a low density, they could be amorphous, they could be crystalline, they could have a hydrophobic character. They sometimes tend to float on top of the liquid vessels. So when you are mixing such powders, and they may not wet easily, you need to have a rigorous mixing so that the powder goes into the vortex, a kind of a vortex, and therefore gets mixed into or gets wetted enough and then gets solubilized. So it is important to understand what is the density of the powder, what is the quantity of powder with respect to liquid, and how easily it can solubilize. So you use the right kind of a mixture. Again, a part of process development and scale up studies. And then there are general mixing arrangements. Of course, you need to have a good mass transfer, heat transfer. You need to, if it's a suspension, you need to keep the suspension homogeneously suspended. So there is a suspension, you know that the particles are floating there, but you need to keep it homogeneous during the formulation and during the entire process till you put it inside the vial or the PFS or the cartridge. Because if that suspension is non-uniform or some amount of settlement occurs, the dose which goes into the vial becomes non-uniform. So from that perspective, mixing is very important, and therefore it is important to choose the right kind of a mixture. This is a table which I've left there. It is coming from one of the vendors of a magnetic mixer, magnetic mixer manufacturers. So you will see that there is also a bioreactor agitator there. They've said what kind of agitators can be used. They could be top mounted, they could be bottom mounted, whatever it is. There are different geometries of impellers which are there. A subject which is another topic altogether. What are the different impeller geometries, etc. Topic of chemical engineering. Any chemical engineers in the room would have studied this as a part of their chemical equipment design, chemical process design. Okay, so I'm leaving this thoughts with you. There are some pictures of some magnet. These are the normal kind of mixers which are used in formulation of an aseptic product, and that's why I've only brought in this images. Of course, I could have brought a library for biomanufacturing and others also, but since they were not relevant to sterile formulation part of it, I only picked up the pictures of those which are generally used in a formulation assembly. So this is something which could be a low shear one. This is a general injectable application. This is a high shear mixture. And this again is something which could be used for powders. Okay, so these are the four examples that I have gotten from a literature of a manufacturer. So with this, we come to the end of this session. I would like to throw open the forum for question and answers, so that we can cover this topic of formulation, and then we will move to the next topic. Of course, I'll call for a break at this point in time after the question and answer session, but it will be a short break. A request that everybody comes very fast so that we can cover the other two topics pretty fast. Yeah, so questions, students. No questions?
Sir, I have one doubt.
Yes, Shashank.
Sir, we place the impeller in the 90 degrees, some aerations will be happen, sir.
Sorry.
We place the impellers in the 90-degree angle, and some aerations will be happen, sir.
90 degree to what?
To the solution.
So you generally do not mount the agitators perpendicular to the vessel, perpendicular to the vertical plane of the vessel. You normally mount the agitators in a vertical plane. They could be at an angle from the zero vertical plane, which is if you take a vessel, and if that vessel was, so let me go to the so normally your vessel axis is this. This is the center of it. You try and place the agitator to the center if it is possible. If it is not possible, then it is possible that you put it at an angle. So you put it at in the vertical, but you still put it in a vertical plane, but never on a horizontal plane. Why you don't put it on a horizontal plane is because of the fact, two reasons. One is that you want to mix every bit of it in a formulation, and formulations are normally that we're talking about are liquids, emulsions, or suspensions. And they being liquids, you need to mix till the last drop of the product is out of the tank. right? Second thing is that if you were to keep it horizontal, then it will create a force only in a certain direction. So when you keep any agitator of this kind, okay, the axis in which it will rotate is this axis. The center axis is this. Now, if this center axis is slightly tilted, then it would be at 15 degree, 30 degree to the vertical plane. Now, when you do that, the vortex which is created is going to be top to bottom. If you place a agitator in this direction, then what will happen is that that vortex will only be here. Now that vortex may not be sufficient to take care of the entire vessel volume, because you want to make sure that you are mixing it till the bottom. The impeller size then tends to go up. There are certain applications where you do have these applications, but they are more in the chemical synthesis plant and fine chemical manufacturing, not in biopharma, pharma applications. Generally, you will see agitators in a vertical plane. Does that
Okay,
answer your question, Shashank?
okay, sir.
Okay. Any other? Okay, so on an aeration, Shashank, what you were telling, what happens is that the agitator will still be vertical, that is in a fermenter. Okay, and what is generally done is that the air is bubbled using a sparger. So there'll be a ring around the agitator. So if this was centrally located, there would be a ring around this through which the gas will be bubbled. And the agitator is designed in such a way, it will not be a single stage impeller, it will be a multi-stage impeller. The agitator will be designed in such a way that the lowermost impeller where the gas is bubbled will break the gas which is being pumped in into very fine small bubbles, so that it can uniformly distribute through the mass of the fermenter. The upper impellers are generally meant for good amount of vigorous mixing, so that these bubbles and the ingredients get mixed, and therefore the oxygen and the nutrients are available to the cells which are being which are growing in that fermenter. So that is the aeration part of it. But even in that situation, you only have a vertical plane of vertical axis in which the agitator is mounted.
Okay, sir.
Okay. Any other questions? Okay, so let's take a short break. I would request that we come back by 10:20, another 7 minutes. In those 7 minutes, please take your bio breaks, come back fast, because the next topic is slightly a longer one, and I would like to cover it. And one more topic from the following session also, I want to try and cover it in the next session. So let's meet at 11:20.
Okay, sir. Thank you.
Thank you. Is a suspension. You cannot filter certain kind of emulsions, or you cannot filter certain high viscous products. Again, you will use filter sterilization as the n minus 1 and step n for doing the sterilization process in situ for in aseptic processing. This filter sterilization is used very well in biomanufacturing also. Remember, we did the UFDF after the ultrafiltration or the concentration is done. Also, you will filter it and fill it into a bag, which is then stored in bulk, and that bulk bag is then brought for formulation that we covered in the morning. And after you formulated it, before you put it into the filling machine where you fill into vials and all, again you filter it. So if you were using a filter as a step of sterilization, then your aseptic process or your sterility assurance levels start from just before the filtration process is incorporated, and before that you just talk about bioburden reduction. So in our sterility decision tree, if a product can be filtered, then the previous steps need not be following aseptic processing techniques. If the product cannot be sterilized and or filter sterilized, there are products like suspensions, as I said, certain emulsions which cannot be filter sterilized. You need to follow aseptic process throughout from start to end. Okay. So sterile filtration does not destroy microbial life. It just removes the microbial life from the rest of the product. Okay. Types of filters which are used in the biopharma industry and the injectable or sterile manufacturing are particle filters, microfilters, ultra filters, nano filters. What are particle filters? The size range is normally 10 micrograms to 200 micrometers. So any particle which is between 10 micrometers to 200 micrometers will get filtered by using such filters or called as particle filters. You have microfilters which are zero point 0.1 micron to 10 micron. Then you have ultra filters which is 0.001 micron to zero point 0.1 micron, and nano filter which is less than 0.001 micron. Now, your question would be that if ultra filter and nano filter are so good, why don't we use all the time that? The problem on using a filter of only one size is that if you had different sizes of particle, the filter with a lower porosity will get choked when the large particles hit it. So generally, when you design a filter sterilization technique, you will create what is called as a filter train or a series of filters which are placed one after the other in varying sizes, so that your filtration process is working in a way that the first filter removes the larger size particles, and the next filter is used to remove the next larger size particles, and then so on and so forth, so that your final filter, which is generally a zero point 0.2 micron or a zero point 0.1 micron ultra filter or a micro filter range of a product of a filter, does not get choked due to other particles which may be larger than the range of particles that they are supposed to filter. Okay, so we are going to talk a little bit about filter train designs, but that is more a product specific topic. And when we develop the formulations, the level at which you have to filter different solutions during formulation has to be decided there only. So you eliminate the particle. One thing that has to be remembered in any process development work that you do is don't try to carry the impurities and the problem till the last stage. If there is a problem in the initial stage, you know that there is an impurity or a particle which is there in the first stage of the process. See if it can be removed then and there, so that when it is not carried forward to the next phase of the processing or the next stage of processing. Sometimes you may not be able to help it because of the fact that you need to kind of have that solution in that format to be able to do the next step. But if you can help it, if there are large particles, let's say you're preparing a tris buffer, you use the commercially available tris salt, and then you create dissolved it in water. At that time you saw some particulate contamination which was there, which was falling within either 10 to 200 micron or zero point 0.1 to 10 micron region. Then you try and put a filter after the tris salt is solubilized in the buffer, and filter it before you use it for the next step. So that is the key to filter usage, that you use the right kind of filters at the right places, so that you do not load the filter at the end. The end filter is more for removing the microbial life. So except for a microbial life removal, it should not be subjected to having to remove the particles, having to remove the microfibers or colloids or yeast which may be carried over. Try and eliminate all those before you get to the final filtration. So this is a key to the filter selection and filter train design. Now, filters are classified as hydrophilic, they wet spontaneously, or hydrophobic, which they do not wet spontaneously. Hydrophilic filters are used to filter solutions and mainly aqueous, while hydrophobic solutions are used to filter gases, solvents, strongly acidic or alkaline solutions. So, applications are very clear. If your formulation was aqueous in nature, use hydrophilic filters. If you are trying to use a gas, like you are using nitrogen or air during the pre and post sterilization operations, use a hydrophobic filter. If there were solvents, sometimes your formulations contain certain amount of ethanol, sometimes certain amount of IPA as a part of their formulation. If that was the situation, then use a hydrophobic filter in such situation. And if you had a very strong acid or an alkaline solution, um, again use hydrophobic filters. Right now, how does a filter function? The filter functions on a combination of three techniques. One is sieving or screening. Very simple. The filter is arranged in a form of a cartridge, or a pleated cartridge, or it is formed in form of a circular or a membrane, flat membrane, whatever it is. When a particle hits a screen, obviously the particle does not go through, and therefore sieving occurs. If the particle orientation is in the direction or that, the sieve can retain it. Simple technique, simple physical unit operation. Entrapment or impaction. So when a product goes into the filter, it tries to go on the other side of the filter, because the liquid is flowing from one side of the filter to the other side of filter. When it enters into the filter, you see the filter is constructed having a treacherous path. So it is not a straight sieve. It is just not like the sieve or the sieve that we use in our residences to sieve the particles in the house. This is more like, yes, there is a sieve effect to it, but it is also multiple sieves put together. So if you took multiple sieves and arranged them in such a way that the hole was not a straight path, but it followed a treacherous path, what would happen? The liquid will find its way out, but the particles which are there, which are larger than the size of the hole, will always stay outside and will not be able to get through. So when you have sieves which are arranged in such a way that the liquid is moving in a zigzag direction, or a Brownian motion is created in a way, then the particle will entrap, because it is not able to move as fast as the liquid. The liquid will just go left, right, center. It will just go out, but the particle will take some time because it needs energy to move, and therefore it gets entrapped. So that's the second mechanism by which a particle gets entrapped into a filter. The third was, of course, the electrostatic attraction, because the particles are generally charged. There is a charge on the filter also, because it is made out of a certain MOC, could be PTFE, could be PVDF, could be cellulose nitrate, whatever it is. So there is a certain amount of charge present on it also, because of the fast flow of the liquid. There is an electrostatic charge which is created. If the charge is opposite to the charge of the particle, then the particle tends to get attracted. So opposites attract each other. So by just that electrostatic attraction also, the particle will get attracted. Normally, microorganisms, the bacteria have a negative charge on top of it. So if you had a surface which was positively charged, the bacteria will go and get attracted to it, while the liquid will just go through. So that is the third mechanism by which a filter causes. It's generally a combination of these all the three factors which causes a good filter efficiency to occur. Now, increasing or prolonging the force behind the solution may tend to sweep the particles. So this is important, that when you design the filter, you have to design the filter area correctly, because when you are pumping, you're continuously pumping. You had 1,000 liter batch. Now you're pumping it through a 5-inch cartridge filter. You have so much of liquid flowing there that if some entrapped particle was there, or some sieve, some material was stuck on the sieve, or there was an electrostatic charge. If there is sufficient flow, it can get dislodged and flow away with the liquid itself. So it is important to make sure that you size the filter right for the quantum of energy that you are going to impart to the liquid to flow through the filter. So this is an important factor while designing it. Now, what are the factors which affect the efficiency of a filter? Obviously, the type of particle. What is the source of the particle? What is the charge of the particle? What is the size of the particle? What is the shape of the particle? Why does shape come into play? If you look at the bacteria, it's a longitudinal globule, right? It's like we saw the picture of it yesterday of an E. coli. So it's like a mycelia or like a long structure. Now, if this bacteria approached in its vertical position, that is on a smaller dimension, and hit a filter, it may go through. But if it hit the filter in its longitudinal section or the other axis, which is the larger dimension, then it may not go through. The sieving itself might work there as a filtration mechanism. So the shape, size, charge, and source are all important factors which affect the efficiency of a filter. The type of polymer or the filter material becomes very important. What is the material of construction of the filter? That becomes important because you have the electrostatic charges which play an important role, including those of microorganism. You need to consider hydrophobic attractions. You need to worry about hydrogen bonding, and of course our zeta potential from van der Waals forces. So those of you who have studied physics or engineering, in engineering, van der Waals forces were a topic which were a subject which was given in importance. Why is this important? Is because depending on the type of material, these forces vary and therefore could affect the efficiency of a filter. Filter membrane thickness, the filter thickness slows the flow characteristic. So simplest thing is, okay, if I have very thick filter, nothing will go through. Yes, that's true. But if you have a very thick filter, it also slows down the flow rate of the liquid through it, and may also affect the particle absorption mechanism. Why? Because as you filter it, now you slow down the flow. When you slow down the flow, what happens? The energy is lost. The liquid also is going at a very slow speed, as and the particle is also moving slowly. They may both be able to get out of the filter. So from that perspective, the thickness optimization is essential, or the way in which the filter is constructed is obviously very important. Porosity, obvious factor, that smaller the porosity, greater the retention. But as I said, if you put a large particle on a very small pore size, it will get choked and therefore the flow rates will drop. So it is important to design the filter train with the right porosities depending on the particle size which you are trying to filter out. Temperature definitely affects the filtration efficiency, because if you had a higher temperature, the Brownian motion of the suspended organism increases. If the Brownian motion increases, what happens? It has a tendency to move very fast and could potentially get entrapped or go through. So the other effect is, if you expand the if you increase the temperature, the pore size may increase. Therefore, the pore holes become more passage wide, and therefore the microorganisms depart. So temperature is important. You try and filter it at normally at room temperatures. All studies are done at room temperatures. Or if your product needed to be refrigerated or was at 2 to 8 degrees centigrade, then you do the filtration studies at 2 to 8. So the temperature is very, very important, because if it's very cold also, the particle is going to be sitting very quietly and is not going to be jumping around. We need certain amount of Brownian motion to capture these particles which are suspended inside the solution. So the doing the filtration at the right temperature is important. Also, another effect is that if you are using oils and polymers, then the viscosity of the product also changes with the temperature, and if you had a slightly higher temperature, you may be able to filter it better and also capture the particles. But obviously you cannot increase the temperature if the product was not stable. Okay. The properties of the fluid itself are important, the viscosity, the density, etc. So, very important as to what you are filtering, what are the physico-chemical properties of the solution itself is very important factor which affects the filter efficiency. The pressure at which you are pumping the fluid, the flow rate at which you are pushing it, and the time for which you want to do filtration, all these are very important factors which are going to uh, which are going to affect the efficiency of a filter. Sorry, I'll just put my phone on silent. Sitting in office, so a lot of. Okay. Now, what are the applications? They can be used for simple particle retentions, or they are used for sterilizing solutions. But in the context of the topic that we're discussing of sterile manufacturing, it is important that there is a particle retention effectiveness. They do not shed any particles themselves. They are non-reactive and are disposable in nature, meaning that it can be disposed of easily. Therefore, they can be handled as a solid waste easily. So these are the properties of a filter which are needed, but they are used for sterilizing the solution. So one of the applications is sterilization itself. The filters are available either as flat membranes or pleated into cylinders or cartridge filters to increase the surface area and thus the flow. So there are various configurations available, flat or pleated. Generally, the ones which are used in formulation and in the fill finish are cartridge filters or cylindrical filters, pleated cylinder filters. They are placed inside a stainless steel housing and then steam sterilized. So generally these filters are manufactured by the manufacturer. They will be sterilized through gamma irradiation or an ethylene oxide sterilization technique. They will be shipped to you. But when you place them inside a housing, the housing itself needs to be sterilized, so you will sterilize it. So after you place the filter inside a stainless steel housing and in a train, you need to make sure that you sterilize the entire setup so that there is no contribution of any bioburden from that assembly. Of course, validation of the sterilization of the filters occurs with usage of thermocouples and spore strips, or your biological indicators, etc. And thermocouples are used to validate the sterilization, which is done by moist heat. Okay, and identify the cold spots within the filter assembly so that it can filter it sufficiently. So generally, filters, whether they are in the air line or they are in the exhaust, or which are both gas applications, or they are used for product or any other transfers, if you are doing an aseptic process, you need to, after mounting it into the assembly, into the housing, steam sterilize it. There are filters which are available, which can be, if which are capsule filters, which can be directly autoclaved and used, or they may come ready to use, which means they are gamma irradiated with the end connections. Generally, you need to connect it to a filter, to a liquid source, right? So you will connect it to a tank, or you will connect it to a vessel, and in or a cylinder or a bottle or whatever it is. In that situation, you need to make those connections aseptically. So you sometimes can use directly gamma sterilized systems, provided that you are able to make end connections aseptically. Now, here is a table which gives the different materials of construction which are used in the industry. The most widely used today are, of course, PVDF and PTFE as two large usage in the aseptic process zone that we talk about. Sometimes we also use polypropylene because It has got excellent chemical and mechanical resistance, but it's a hydrophobic material. So because it's a hydrophobic material, we need to use it in applications. Now, you would ask me a question: when PTFE and PVDF is a hydrophobic base, why do you use it for product filtration? Because that is what it is. It is a hydrophobic base, but it is generally surface treated chemically to make it hydrophilic. So this is a technique which is used in PVDF filters. So though the base material is hydrophobic, you make it hydrophilic by a special chemical treatment. So you have to do studies of compatibility of the different material of construction of the filter with your product, and that would be how your filter train gets designed. So this is the topic on filter selection and filter train design. So tests must be performed to demonstrate that the product does not affect the retention properties of the filter, as is accomplished in most cases with bacterial retention studies. So what is generally done is that you do a bacterial, prepare a solution with bacteria, known bacteria of a certain load, and you just put it through the filter to make sure that the bacteria is getting retained, but any other salt or any other product which was there is passing through without being retained on the filter. Because if your product starts to get retained on the filter, then that filter is of no use to us. Okay. We need to perform the test of leachables and extractables. Very, very important that this leachability and extractables are definitely something that you need to study and make sure that when you are trying to put the product through that filter, it does not lead to any contamination because of any extractables or leachables. Okay. Now, normally there is one statement I'm making here, which I brought in because it was important, that though the filter manufacturers will give you compatibility, extractability studies, leachability study results from their side, the ultimate responsibility lies on the manufacturer of the final product. So you can use the data which is provided by the filter manufacturer, and then you may have to do additional studies to make sure that the interaction with your product, because your product may not have been studied by the vendor who's manufacturing the filter. So it is important to carry out a extractability and leachability study with the material of construction by taking these filters from the product manufacturer, from the filter manufacturer, and subjecting it to your own product. So please remember that when selecting a filter, please ask the vendor to provide you extractable leachable data, compatibility data with various solvents, and then ask the people who are doing the formulation development that they have done the extractability studies, compatibility, extractability, and leachability studies. Okay. Now, what is the other data that you require from the filter manufacturers? You need to require what is the first volume required to yield a negative oxidizable substances. What does this mean is that what is the amount of liquid? When they manufacture the filter, there are certain oxidization agents which are used in the filter manufacturing process. Initially, when you use the filter, you need to flush it out completely using WFI or the gases, and in that situation you need to ask them what is the minimum flush volume required to get to yield a negative oxidizable substance, so that no oxidizable substances are coming out on the other side. What is the level of extractables, leachables? And when we talk about extractables, we talk about oligomers, mold release agents, antioxidants, wetting agents, plasticizers, manufacturing debris, membrane backing, cartridge body, and O-ring material, because the cartridge, when it is made, comes with the O-ring already fitted to it, and obtained with different solvent exposure. So ask for the extractable data. Limits of flow rate, temperature, and pressure. What is the maximum flow rate you can subject it to? What is the maximum temperature? What is the pressure? What is the minimum flow rate? What is the minimum temperature? What is the minimum pressure? What is the sterilization temperature, and at what time of sterilization have they validated the filters. So all this data is generally available from a good filter manufacturer, and therefore this data has to be taken. It has to comply with 21 CFR 210.3b criteria of no fiber release. Procedures for filter sterilization, they generally give you this along with the data on how many sterilization cycles the filter can withstand. They would have done these filter validation studies. So they provide you with a validation document for the filter. The filter bubble point or diffusion rate for the in-process integrity testing. So we are going to cover integrity testing as a topic today, and in that we need to understand what is the bubble point and the diffusion rate, and these are generally given by the filter manufacturer. Correlation of the integrity test value and the amount of B. diminuta retained. So B. diminuta is a bacteria which is used as the one of the microorganisms to do the bacterial retention studies, and to say that at what integrity testing value what is the correlation between the retention of the bacteria that we're talking about to the integrity of the filter is a correlation study that they might have done. I'll cover a bit of this in the next segment or the next next presentation. But also what is important is that there are this data is maybe of importance to start with, but today with the PDA technical report 26, which also we are going to touch upon a little bit in the next presentation, there are certain things that this correlation just is not enough to certify the integrity of a filter. And written instructions and specifications for doing the filter integrity. What is the recommended procedure for doing the filter integrity testing? Why this filter integrity is important, I'll come to it. So this is where it is. It's a non-destructive physical test which assesses the presence of oversized pores or other defects that might compromise the filter retention capabilities. So what is the test? It is a non-destructive test. You either measure the bubble point or the diffusion, or you could do a water intrusion test or pressure hold test, and I'll come to the basic definitions in this presentation in more detail in the next one, where you just do a check that the filter is not damaged. In other words, you are making sure that the filter, because it is pleated, it is formed into a cartridge form, it has got a certain thickness. How will you, and it's microporous, right? It is zero point 0.1 micron, 200 micron. You can't see it with your naked eye. So, how do you test to make sure that it is not defective? For doing that test, we do what is called as an integrity test of the filter. Okay, there are certain regulatory requirements around the integrity testing of the filter. The EU GMP Annex 1, the latest one, which was released last year, mandates that the integrity of the sterilized filter should be verified before use and confirmed immediately after use, using methods like bubble point, diffusive flow, or pressure hold test. So this is a requirement which is defined by the regulation, that you need to do pre and post use filter integrity testing. So to make sure that when you filtered the product, before you filtered the product, the filter was fine. After you filtered the product, also the filter did not get damaged during the process of filtration. So this is what the EU GMP is asking for. FDA guidelines emphasize post-use integrity testing. So that is done outside, meaning you, after you finish the process, you take the filter out, you do an integrity testing, and this is a process that you will do 100%. The pre-use is what is being debated currently in the industry. Though it is a part of the EU GMP Annex, there are certain ifs and buts around it, and people are not comfortable to do a pre-use, because you use a certain solvent and then after that you do your product. So there is a little bit of a question mark there on how to do it without affecting the stability assurance. We will probably touch upon this, but I'm not going to deep dive into this topic, because it's a deep topic which we will need to touch upon when you actually do the manufacturing processes, and not at a level where we are trying to understand the concepts. Okay, so very important that you have to understand that after usage of a filter, every regulation mandatorily asks you to test the filter to make sure that the filter was not compromised during filtration process. So after the process is over, you immediately take the filter out and subject it to integrity testing and make sure that the filter was integral before you throw it out. And this is a report that you have to attach with your batch manufacturing report. Without this, your quality assurance will not give you the approval to dispatch the product. PDA technical report number 26 recommends routine pre-use and post-sterilization integrity testing of filters. So PDA is Parental Drug Association, which is a worldwide organization working specifically on injectable product subject, and they have their own technical studies and reports which are conducted, and when these are conducted on a day-to-day basis, then you definitely have to follow some of these studies because they have interesting findings which have come. Okay, so PDA is not a guidance, it's not a regulation, it's just a industry body which is what we need to make sure is studied, because there is a lot of data which is available, and yes, it holds weight when the regulators look at it. Okay. Methods of integrity testing. Bubble point test measures the pressure at which the liquid begins to flow through the filter. So when you subject a filter to a bubble test, what you do is you allow the filter to pass through it and then you pressurize it. The moment that bubble starts to form and come out into the other side of the filter is where the bubble point test is. Okay. So the bubble point is when the liquid starts to flow through the filter. That instant at which, or the pressure at which the liquid will start to flow out is called as a bubble point. Diffusion test evaluates the diffusion of gas through the filter. We'll get into a little more depth about it. Pressure hold test checks the filter's ability to retain the pressure, just like we did pressure hold test of autoclave and the vessel. Here, pressure hold test of a filter to make sure that the pressure is being retained by that entire setup. Water intrusion test determines the filter's resistance to water intrusion. So you try to pass a water and it should not go out on the other side. It holds. That means that the filter is integral. So that's a water intrusion test. Now, best practices, manual bubble point test. This method is essentially used for assessing filter integrity, and water intrusion test gets affected by the filter type, weighting agents, and the temperature. So integrity testing ensures the reliability of filters in critical processes. And if you encounter any issues, troubleshooting integrity test promptly is crucial. Excuse me for a moment, please. Hello. 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 Okay. Now, I'm going to just bring a concept of sterile filtration validation. Okay. This is what is a sterile filter master plan. So integrity testing, as you see, is an important aspect of it. Retention is an important part of it to prove that the bacteria gets retained onto the filters. Extractable leachables and compatibility are both important. Binding proves that filter does not remove stream of components. It does not absorb the component of interest which you are trying to filter out. Right? You're filtering a solution. If your product starts to get bound onto the filter surface, you will have a problem. Stability proves that the filter does not affect. Again, stability is important, that it does not influence the product itself. So sterilization, that you are able to effectively sterilize the filter without compromising the integrity of it, and fit for use prove that the filter meets all the requirements within product and process conditions. So these are the 8 elements of sterile filtration validation. Now, the highest focus actually stays on the first 3 parts, which is retention, extractable, leachables, and compatibility. When you are choosing a filter, choosing the right filter is based on these 3 factors more than the rest of it. Of course, stability or not of compatibility is where you will do those studies, but These are the three most important factors that you will check. Risk assessment for a fluid path compatibility evaluation strategy. Look, you have to know at what stage of the process are you using the filter. Are you using it in upstream, downstream, or final phase? Of course, the if the compatibility is not there, the risk is highest in the final fill, and the downstream and upstream you still have opportunities to correct any leachables or extractables or any filter media which gets into your product. But when you're in a final fill, you don't have that opportunity. So the risk is highest at the final fill, then downstream, and the least at upstream. Drug product, based on the type of drug administration and drug development process, the compatibility evaluation has to be done. The filter has to be defined right at the stage of the formulation, pre-formulation stage itself. You cannot say, okay, I've developed the formulation, now I'll decide what filter I'm going to use. The filter selection, the media, whether it is cellulose nitrate, polyether sulfone, PVDF, what size, what porosity is done right at the time of pre-formulation or formulation development. Okay. What is the contact surface? What are the different MOCs? These are important from a perspective of choosing the filter type. What is the contact type? What is the surface area? And of course, supplies data on similar conditions or documented historical data. So we spoke about biopharmaceuticals. We have Genentech. We have Eli Lilly. We could have Biocon, Sun Pharmaceutical, whoever is the innovator of a product, they would have used a certain type of filter and under similar conditions for the same product if you are a generic manufacturer. And using that data becomes important, or having access to that data sometimes is important, because if they were using PVDF for a product, they have done sufficient study for you to screen your the filter that you have to use becomes smaller, because now you have to maybe say, if the innovator was using PVDF, I can probably use PVDF myself. You of course have to do the studies, you have to prove the validate, you have to select the filter correctly, but then your search window goes much more shorter, right? Now, you always have to test the filter with the drug product. You have to define the parameters at which you are going to run this, and make sure that the filter is compatible with the product that you are trying to filter at the given temperature and for the contact time that you're looking at it. So this is the other important part that you have to do. So what are you doing is, first you are looking at retention, extractable, leachables, and compatibility by doing interaction with the vendor, doing some literature survey. Then you are of course trying to establish at which all stages you are using, and do the compatibility evaluation strategy, that what am I going to measure, how am I going to measure, at which stage I'm going to use the filter. Then you are doing the actual test with the drug product. Now, what are the requirements on terms of retention? You have to define. These are some of the guidelines which are provided by FDA. It is vital that laboratory experiments are carried out to simulate actual product conditions, pH and viscosity of the material to be filtered, flow rates, pressure, temperature, compatibility of material with the filter itself, and the effect of hydraulic shock are factors of production which can affect filter performance, and which should be looked at during the filtration itself. Just one sec. Anirudhji, good morning. Anirudhji, good morning. Sir, can I call you? Good morning, sir. Good morning, sir. Sorry, students. It was an important call. Okay. Okay. Now, And the goal of bacterial retention validation study is to have documented evidence demonstrating that the filtration process will consistently remove a high level of standard bacterium or isolate under the process conditions. So these are already guidelines which are defined in the 2004 guidance documents for sterile product development, and therefore the retention experiments become important. So what is generally done is that in the initial stages, you define the material of construction, take small size discs, circular discs, just like your Wattman paper, which you used to get for your laboratory experiments, and do some flow studies on that, saying whether my material is compatible, whether I'm able to filter the product, what is the flow rate I'm getting per unit area before the filter chokes. So a lot of those studies are done. Then you actually take the filter of a smaller size. And most of the filter manufacturers will support you by giving you different sizes of the filters which are scalable. So you take a disc which is like a circular paper which you put on a Buckner funnel, do your test to check compatibility, flow rate, etc. Define your area. Then you take a smaller size cartridge, maybe a 2.5-inch cartridge or a 5-inch cartridge or a 10-inch cartridge, depending on the volume of the R&D fluid during the process development or product development. What is the quantity of fluid you have? You do the studies. First, you do it under non-sterile conditions to make sure that your filterability is achieved, your pH, viscosity, temperature. If you give it too much of a liquid, then at what rate it chokes. All these studies are done in the laboratory. And you have to have documented procedures for this to prove that how you have taken the filter. You can say that I started using PVDF because there was a reference, earlier reference to it, and I got it from a literature survey, but you still have to do these studies. And then, of course, you need to do the bacterial retention studies, because the ultimate goal in sterile manufacturing is to get rid of the bacteria and say that your filter sterilization process is effective. So from that perspective, there is one which is the product interface, and the other one which is the microorganism removal, which you have to worry about. Okay, so I think this is the end of the. Okay, so this is what is the drug test, that you can take the actual drug product, inoculate it with the challenge organism, give it a certain incubation time, and then you say whether the filtration is working out. Then if it is non-inhibitory, then you can directly go ahead and use the filter. If it is not, then you need to worry about the scenarios in which you have to filter it to remove the microorganism. You always do a study with a worst case condition. So you look at the drug product and say, what is the maximum viscosity? What is the lowest temperature I'm going to do it? So the process parameters and the drug product properties of the worst case are taken. You choose the actual product and the highest process parameters. The lowest temperature for filtration will give you the least flow rate. Higher temperature will give you the higher flow rate. So you will try and filter it at a lower pressure, give it more time and see whether the filtration is okay or not. Sometimes you'll do too fast filtration to see if the bacteria is getting retained. You will also look at the filter structure and bioburden. So all these four factors that you are talking about have to be taken in their worst case scenarios and tested one by one. So you test one at a time first to establish the worst case scenario of the process. For that worst case scenario and the best case scenario, now you challenge it with bioburden. Then once you've done that, then with the combination of those two, you change the parameters of the different type of filters, different type of physico-chemical properties of the drug product itself. Okay, so what are the worst case conditions? Generally, this is just an example that I could find, so I thought I should bring it to your notice. This is not necessarily the Bible, but it's something which can be used. So, if the osmolarity, the main effect is the size of the organism, worst case value is the highest osmolarity will affect the size of the organism, makes it the smallest. Surface tension is lowest. Retention mechanism is what gets affected if the surface tension is very high. then you are going to have a good flow path. But if the surface tension value is at the lowest, it becomes a worst case scenario. pH 5 to 9, highest filter pH being at the highest to the lowest rate will affect different parts. So organism proliferation at 5 to 9, filter compatibility when the pH is high, and when it is lowest or the highest, retention mechanisms can get affected. Ionic strength can also affect the retention mechanism. Retention can also, viscosity can also affect the retention mechanism. So higher the viscosity, maybe higher the retention, but also lower the flow rate. Process parameters, flow, if there is a high flow rate, then you could have effect on the retention. Things may easily pass through. If the filtration time is too long, then you may have a growth through bioburden, meaning that the bioburden starts to be retained, but then there is enough time for them and the temperature conditions are right for them to multiply, because remember, you're not killing them, you're just retaining them. And then if there is enough of those microorganisms, they may fall smaller spores which may go through your product. So it is important to keep the filtration time optimal. Hydraulic shock, if you do hydraulic shock, if you give a hydraulic shock to the filter, it may just tear, and therefore it may blow through. And temperature, of course, the bioburden proliferation and membrane compatibility, as well as the pore size expansion, becomes the worst case. So you are going to test the filter in the worst case scenarios and define the parameters at which you are going to operate the filter and which filter works the best for you under these worst case scenarios, and choose them without affecting the product properties. Okay. Uh, Obviously, when you're doing a cold method of sterilization where you are not killing an organism, the regulators are worried, because it is not a sure method of killing a microorganism. It is just a chance that the filter will retain it, and therefore, what is important? Patient safety is more than satisfying the regulators. So patient safety becomes very important. Make sure that your product is suitable for the patient. Regulatory aspects, what does this mean is that there are regulatory inconsistencies. If you look at through various regulatory guidelines, you refer to you refer to US FDA, you will find somebody says you should do PubSet, somebody will say, meaning pre-use and post-use. Somebody will say no, only post-use is okay. Somebody will say bubble point is acceptable. Somebody will say water intrusion test is acceptable. So there are regulators which are not harmonized. But what is important? Is my patient going to be safe? That is the first point that you have to worry about. Traditional challenges can be overcome. So what has to be done is that when you define the process, a lot of times we used to feel, okay, I can't use a solvent based product, or I can't filter in emulsion, so I'll have to sterilize the whole bulk of it. Today filters are available which can do such challenging tasks, and therefore you have to make sure that you find the right solution to it. Life cycle effort is very important to make sure that you look at the life cycle of the product, and take your decision on what kind of filters that you check. And last but not the least, you choose the right filter manufacturer who has sufficient data, can give you support if you wanted to do extractability studies, leachability studies. Please remember that doing these studies is not easy. You need means mechanisms, and you need a lot of other what do I say, analytical methods to arrive at the extractable and leachability studies. In such a scenario, you need to make sure that you need to make sure that you have the right partner who can support you. You choose some fly-by-night operator who's supplying filter in the market. You will go to the market, you will find a lot of filter manufacturers. Choosing the right partner is very important here. So with this, I will come to the end of this question and answer session. Um, I know I have taken all the time which was allocated to this session. So let me open this session for a quick question and answer session, and then we will take a short break and again come together. So any questions on the filter sterilization and how you select a filter and how don't ask me questions right now on the filter integrity testing, because once we cover it in the next topic, you will get more information, and after that you can ask me of course questions at that time. Questions? Okay. So if there are no questions, let us take a short break. Again, not more than 10 minutes. So we get back at 11:25 to this session, because the next session has three large topics to be covered, and we will need time to discuss that out. So I would request all of you to be back by 11:25 latest. It is 11:16 right now. Okay. All right. See you in the next session. Okay, is everybody back? Can we start? Dr. Vishal? Can someone please confirm that everybody is back?
So as all of them are came.
Okay, so let's start. We are going to cover three different topics in this lecture. Incidentally, this lecture is the last of the lectures that I'm going to deliver to this batch. However, we have three very important topics which we are going to cover. Since we talked about filters, we will talk about filter integrity testing and filter validation and cover some aspects of the PDA Technical Report 26. We will also talk about what is called as media fills or aseptic process simulation. And then the last but not the least, the aseptic techniques and behaviors which affect an aseptic processing in the manufacturing setup. So filter integrity, we talked about it. We said it's a non-destructive physical test to determine the presence of oversized pores or defects that may compromise a given filter's retention capability. Plays a very important role in making sure that the filter was reliable and efficient in doing a sterile filtration process, and in doing a filter integrity testing ensures that the membrane filters retaining their intended functionality, specifically in preventing passage of bacteria and other contaminants, thereby preserving the integrity of the entire filtration process. Because you are not destroying the microorganism, it is very important to make sure that the filter did not fail, and therefore bacteria or any other contaminant did not sneak through onto the other side of the filter. The users and filter manufacturers perform the filter integrity test to validate the filter performance. Such evaluations include parameters to assess the ability of bacterial retention and ensuring that it adheres to the necessary standards for designated application. So the filter challenge test is done while selecting the filter and the make, type, material of construction, porosity, but you need to repeat some of these tests periodically to just make sure that if there is a change in the microflora of your formulation, then your filter is still effective. On the other side, the integrity test, which is a non-destructive test, is done after every batch, especially on the product filter, to make sure that your filtration process was not compromised. If you find that the filter has failed on the integrity testing after you have, let's say, you formulated the product, you filtered it and you filled it into the vials, and then you took this filter out and subjected to integrity testing and found that the filter was failing, then you have to probably throw the entire batch. So very, very important that filter integrity testing is done in aseptic processing. This becomes a very, very critical factor. The new EU annex, which has been published last year, insists on pre-use and post-use, saying that after you sterilize the filter and before you start to filter it, you need to check the integrity. And after you have used it, also you need to check the integrity. So you check the integrity of the filter before use and after use, and that's called as PubSet, pre-use, post-sterilization, pre-use integrity testing is PubSet. And PubSet is something that you will find as a terminology being used in the industry, saying we need to implement PubSet, we need to implement PubSet. Water integrity testing or can be done in situ while the filter is installed in the assembly line, while the rest of the tests like bubble point test and diffusion test have to be done by removing the filter. Okay. Now, understanding the role of it, we will also cover that there are destructive and non-destructive methods of testing. In a destructive testing, you intentionally alter the structural integrity of the filter, providing tangible test results. This method particularly useful for understanding the filter's limitation and failure point. So you will carry out a destructive testing to check the limits of the filter. So you will put too much of pressure. So if your vendor has said that your pressure cannot exceed 2 bar, you will go all the way to 2 bar or slightly above 2 bar to see how does it affect your thing. In that situation, you might end up destroying the filter, or if you subject it to such a high pressure, then you can't use it in the process after you've done this kind of a testing. That's why it's called as a destructive testing. Same thing for temperature, same thing for viscosity, etc. So those are destructive methods of testing, but they are essential when you are selecting the filter. On the other hand, non-destructive is without altering its composition, you are performing the test. So it is important to do during a routine process non-destructive test. Destructive test will be done initially when you are selecting the filter. Non-destructive tests are done when you are actually doing the routine process. Okay. Key factors such as flow rate, integrity of the filter housing, and measurement of the largest pore size play crucial roles in determining the appropriate testing methods. Some of this data may be already available from the vendor to you, and in such a situation, you could rely on this data if the partner was properly evaluated. Remember that in pharma, even if there was a large firm like, let's say, Millipore or Sartorius, whom you are working with on filter, you still need to perform or do what is called as a vendor assessment to make sure that you have chosen the right partner for your project. But as a name, okay, so destructive versus non-destructive, the differences, destructive will be done more towards a filter selection time, and non-destructive is done on routine. However, if during a routine non-destructive test method fails, you may use one or multiple non-destructive test methods, and then you may have to do an investigation of why the filter failed, may have to subject it to destructive testing methodologies. But what you will normally hear is integrity testing using non-destructive testing methods. These are some of the regulatory guidelines around the integration. So EU GMP Annex, which is there. This clause is retained in the current guidelines also, that the integrity of the sterilized filter should be verified before use and should be confirmed immediately after use by an appropriate method such as bubble point, diffusive flow, or pressure hold test. The integrity of critical gas and air vent filters should be confirmed after use. The integrity of other filters should be confirmed at appropriate intervals. Very clear that if you use it for product, do it before and after the use, immediately before and after the use. If you are doing it for the critical gas or air vent filters, it should be confirmed after the use. And if you are doing it for any other applications, intermediate filtrations, then you can do it at a interval which you have established and have validated. Now, FDA guidelines, which were established in 2004, it says prior to processing and should be routinely performed post use. So it says you test the filter before use and routinely once in a while do it post use also. Now, this is where the problem occurs that if you are designing a facility where the product is going to multiple countries, you may have a country which is located within the EU region, you may be selling it to US, you may be selling it to Brazil, you may be selling it to China, Japan. The different guidelines may have contradicting statements on integrity testing. So how do you design your process? That becomes an important question. Generally, the approach that most of the people take would they would go to the worst case scenario. In the worst case scenario here is that if you look at EU, it says pre-use, post-use, you do it. So whether you even if you go to FDA, it is saying pre-use, post-use is routinely you have to do post-use. You are saying I'm doing it every batch, so they should not have an objection to it. But that involves certain modification in the methodology of your processing time taken for doing the aseptic process batch. Also, there are certain risks involved in doing pre and post use. So, in such scenarios, you have to evaluate carefully, and of course, there is a cost. So given all these, when you are choosing the strategy of integrity testing, it should be carefully evaluated from a regulatory stand perspective and to make sure that you are compliant to the regulations which are there. Now, PDA Technical Report 26 uh, uh, also talks about integrity testing prior to and after use. If one filter has been validated to achieve sterilization with a specific product, then the single sterilization filter must satisfactorily pass integrity testing before and after use. So, what does this mean is that this technical report is a document. I'll probably send it out to Dr. Vasan to distribute it to all of you, or you could just type PDA technical report technique number 26, Google it out, and you will find that this report is available. It's a PDF document. When you go through it, you will find that this is a report which has been created by this association called PDA and is talking mainly around the filter integrity itself. What are the different conditions? What happens in case of a failure? Is the only reason for they are talking about the reasons of failures, and while the regulations have certain reason of failure or one or two reasons assigned within the regulation, PDA actually explores to a much higher depth. And that is why it is important to also take cognizance of this report, because it gives an insight into it. I have brought in some elements of from this report into the presentation, so we will definitely discuss it. Now let's look at the bacterial challenge test and its correlation to the integrity test. Remember this we discussed in the last meeting, that you need to have a correlation between the bacteria, a load of bacteria, and the integrity of the filter itself. And this is generally a test which is done by the manufacturer of the filter and generally will have the data. But in case they do not have a validated data on this, then you may want to do this in your own setup. The physical integrity test is only meaningful when it can be related to a specific filter retention characteristic. What are you trying to do using filter sterilization? You are trying to make sure that the filter is able to retain the microorganism, and Bacillus diminuta is one of the challenge organisms which is used. The reason why, sorry, Brevidomonas diminuta, it's not Bacillus, it's at Brevundimonas diminuta. It's a diameter of zero point 0.3 to zero point 0.4 micron. It's a length of up to 1 micron and a concentration level of greater than 10 to the power of 7 CFU per centimeter square of an area. So the reason why this organism is chosen is because of its size, because it has a diameter of zero point 0.3 to zero point 0.4 micron. So if you were using even something like zero point 0.1 micron filter, when it approaches the filter in the right speed and direction, it is possible that in the diametrical space and in its dimension of diameter, it may be able to pass through. And the length is only 1 micrometer, so it's a really small microorganism. And therefore, it is the most suitable one which is used for bacterial challenge, so that it can give you, if this is retained, and most of the standard flora which is available in any manufacturing environment is likely to be retained. The second thing is that it can grow and proliferate to 10 to the power of 7 colony forming units per centimeter square of an area of a growth media. And in filters, you have very small sites in which the bacteria gets lodged, and therefore this concentration level which it can grow to can help you determine whether there is a proliferation while it is getting filtered itself. On the other side, you will challenge the filter with such a high concentration of the organism itself, so it's like a loaded solution spiked very heavily, so that you are 100% sure that your filter is going to work for you. Okay. Since the bacterial challenge tests are destructive, they cannot be used on filters intended for production use. Obviously, this is something that you do a type test, meaning you choose a filter catalog number, a filter model, you subject it to bacterial challenge test, and then you throw that filter after you've established the correlationship. So let's look at the integrity testing values and this thing, the diffusion of the liquid through the filter in ml per minute is where we are talking about. So as I said, there is always a safety margin. So this is where you are plotting the diffusion rate, and this is the sterile boundary, because up to this thing, whereas the diffusion of ml per minute, which is happening up to this capacity, you are still in a sterile region. If it exceeds these, you become a non-sterile region. So what is done is that your sterile water, you have a high concentrated solution of the bacteria, you pass it through the filter, and then when you're passing it out, on the other side you have these analytical retentive filters. These are filters on which the solution which is filtered from a filter cartridge get retained, and then you plate these filters on a growth media to see whether the bacteria came on this side or the other side. Now, what you will do is that you will also perform an integrity test by diffusion mechanism, and you will see at what concentration of the and flow rate of the water and the bacterial challenge test solution, what was the diffusion rate, and therefore whether it was within the sterile boundary or whether it fell outside the sterile boundary. So you do a bacterial challenge test by measuring both the diffusion rate of the filter as well as by challenging it with a microorganism and correlating it. Till PDA technical report 26 was published, this was data which was used by most of the people and said, okay, if I'm having a diffusion rate of so much, I'm still in a safe margin, I'm still in a sterile zone. But then the PDA technical report came out and it said that no, this is not the only correlation that you have to look at. This is an important correlation and you can look at, but there are other factors to which you have to consider when considering a filter being integral. So we will look into that part also, but this is how the bacterial challenge test is done. Methods of integrity testing. What are the different methodologies by which you can do filter integrity testing? There is bubble point, diffusion pressure hold test, water intrusion test. Okay. This we covered in the last session, but now I'm going to get into a little bit more detail about it. So bubble point test is a test where the wetted membrane filter is assessed to the point at which it allows the gas to pass through under increasing pressure. So what you do is you wet the filter. Now you start passing the gas at a certain pressure through the filter until you see the bubble coming out. So the bubble may not come out when you initially start at a certain pressure. As you increase the pressure, you will suddenly see a bubble coming out, or the bubble will start to flow from a path. As the pressure increases, the smallest pores open, enabling a gas flow and create bubbles. The pressure at which this occurs is known as the bubble point and correlates with the filter's pore size and the surface tension. Because when you wet a filter, there are two factors which are under thing. One is the pore size itself, which will allow a certain size of a molecule to pass through it or a bubble to pass through it. And the second thing is that when you wet a filter, it creates a surface tension, a small curvature on the top of a water glass that you see. Now that surface tension also needs to be overcome for a bubble to go through, right? So this bubble point test will correlate the filter pore size and the surface tension to the pressure at which the bubbles come. Key to reviewing filter's integrity, the bubble test provides a quantitative measure of the filter's ability to resist pressure and maintain its structure. This method aids to identify the potential issues such as blockages or structural damage impacting the pressure drop and gas flow. Consequently, by leveraging the bubble point test, user can ensure reliability and efficiency of the filtration process. So why is this important? Because it gives you the ability of a filter to resist the pressure and maintain its structure without compromising the structure. Up to what pressure can it resist? And if you do this bubble point test on a pre-use, if you have the data pre-use and then or from the vendor, and then you do it post-use, you will see whether the bubble point varied. If the pressure at a lower pressure, the bubble occurred, then you know that there is a structural damage or there is a alteration in the membrane structure, which could have compromised the quality of your product. Now, the next test or the next type of test is diffusion test. The diffusion test is also referred to as a forward flow test, focuses on diffusive flow, measuring how gas pass through a wetted membrane under controlled condition. So in order to achieve this, what is done is that this is normally performed on filter with very large surface areas. And the crucial parameter here includes the flow rate. So how this occurs, I'll just show you in through this. So there is a test pressure. It is dependent on Henry's law. The solubility of gas and liquids is dependent on the pressure and temperature. So when you apply sufficient amount of pressure, the gas is able to diffuse through a wetted membrane and pass on to the other side. And this is what you are going to measure: the differential pressure on the front side, which is the test pressure, and the atmospheric pressure at which it comes out, and you measure whether the gas is able to pass through. And that is the diffusion through the liquid, which is what you are measuring during a diffusion test. Again, how is this important? If you have a diffusion test value pre-use, post-use, when you do this, if the pore has opened up or it has enlarged, then the diffusion will occur much faster at a much lower pressure. So by doing that, you are able to establish whether the filter integrity was compromised. Pressure hold test. The pressure hold test focuses on the stability and endurance of the filtration system. Filter has to be held at a specific pressure, and that pressure has to be sustained over a period of time. If there is no pressure decay, then it means that the filter was integrated. So this is simple like our vessel pressure test or autoclave pressure test that we do, pressure hold test. But this is not one of the most widely used methods. We use either a bubble point test, diffusion test, or more importantly, we use water intrusion test. The water intrusion test is a fundamental method in filter integrity testing, particularly for evaluating the performance of hydrophobic filters. It can be used for it cannot be used for hydrophilic. Why? Because the water would wet the filter and would pass through. So water intrusion test is a very important test used for hydrophobic filters. If you were using PTFE filters for your product filtration or modified PVDF in terms of having a hydrophilic chemical treatment, then water intrusion test can be used to assess the integrity of the filter. How does it work? Test water is applied to the filter and the rate of water flow through the filter is assessed, and this water intrusion test can be performed in situ. Of all the tests that we are talking about, bubble point, diffusion test, water intrusion test can be performed without dismantling the filter from its assembly at the site at which the filter is installed itself. You don't have to remove the filter to do this test. So the water intrusion test is a test which can be performed in situ, while the rest of the tests have to be done by removing the filter. Something to do with motor. I'll come and meet you after this session. Okay. So principle of bubble point, you have a wetted membrane, you are putting a compressed air, surface tension and capillarity effect resist the liquid expulsion from the pores initially, and then when you apply a larger pressure, liquid forcibly removes from the largest pores first, resulting in bulk airflow. Please remember that if there is one breach on the wetted membrane, then the gas will immediately flow through that, and therefore you are measuring that pressure at which the flow is not happening. Now, so you would ask me that we just spoke about diffusion flow where certain amount of gas will diffuse. Yes. So bubble point is a point beyond the diffusive flow itself. So when you are increasing the flow rate or increasing the pressure here, what happens is that the diffusive flow keeps on increasing as you increase the pressure, and that may still be within the acceptable limits. But at one point, it will breach the pore, and there will be a huge amount of flow, suddenly a whoosh flow coming out of that. And that is the point where your bubble point has occurred. So that pressure and at that flow is where your bubble point region is. Compared to a pre-use filter data, you can define whether it occurred at the same position where it was at a pre-use, then the filter is integral, otherwise it is not. This is an equation which is used for calculating the bubble point. Okay, diameter of the largest point. You may either know, you may not know all the factors. You may know the surface tension of the wetting liquid. The contact wetting angle between the liquid and the membrane may be known. The correction factor for the shape of the largest pore, something which is defined by the membrane manufacturer. Differential pressure at which the largest pore will open is something that either you will apply or you will know this, and you will determine whether the diameter, what was the diameter of the largest pore. So P and D are proportional in the sense that you proportional through this equation. So you could determine the D or the P from by using this equation if the one was known to the other. Okay, so this is an equation which you will use generally today. Most of the integrity testing machines are automated. They have the formulas built in. What you have to do is just to buy the right kind of integrity test equipment, put the filter in, and choose the recipe, whether you want to do bubble point test, whether you want to do diffusion test, or whether you want to do water intrusion test, and the system will do its calculation and give you the result. These are equations which are the algorithms which are built into those equipment. When we were doing work in 30 years back, there were no filter automated equipment, so we had to use these equations. Okay, principle of diffusion we have already discussed. Again, a formula there. Again, the pressure or the diffusive flux can be determined by this, or even the thickness of the wetness layer is generally known because this is a data which is given by the vendor. Contact area between the liquid and the gas is also known. Solubility coefficient of the gas and diffusivity of the test gas through the vacuum filter are generally also well established depending on the type of gas you use. The only two parameters which are not known is the diffusive flux and the pressure differential. Both of these can be determined using this method and the equation which is given here. Again, as I said, already built into the integrity machine. Now, When you talk about a hydrophilic filter, then the best method is to use a bubble point or a diffusion. In case of a hydrophobic filter, you use water intrusion test. So water under pressure is forced to intrude into the pore matrix. The greater the overall porosity, the higher the rate of the intrusion. And the water vapor travels from the leading edge downstream through the filter membrane. So basically what happens is that you are trying to force water through a hydrophobic membrane. The membrane will not wet. It will not form a surface tension there. There will be some amount of surface tension formed, but it will not be as prominent as it is in a hydrophilic membrane. You will still see the water breach and go onto the other side, either in form of a vapor or wetness coming out on the other side. And that's your pressure at which you will say that the water intrusion test. This is how it is performed. So as I said, this is in situ. You have the filter installed within the housing, and then you do this test. In the other case, you have to mount it in a certain kind of a special housing and then perform the test. Water under pressure evaporates through the membrane and causes the increase in the headspace volume of the filter. So when you fill the liquid, you apply a certain pressure. When you apply a certain pressure, when this water breaches and converts into water vapor and goes into this, what happens is that the head space volume increases because the water has gone through and the resulting pressure drops. So how do you measure this? That when you have a pressure gauge here, you know what is the headspace pressure. When water intrusion occurs, the pressure drop occurs, and that is what gets converted into water flow rate. So this is the formula for a water intrusion test. A very simple formula: test time, test pressure, intrusion in ml per minute, and the this thing. Now this is where it is directly correlated to the bacteria challenge test as per ASTM F838. Okay, so water intrusion test is what is widely used in the industry today. Apart from bubble point and diffusion, water intrusion test can be done in situ, and therefore it is a preferred method for most of the applications that we talk about. Now I'm going to touch upon this PDA technical report 26 because it brought in the concept of how the filter failures can occur. If a sterilizing filter fails in integrity test, it could be damaged, but there may be other causes for failure that include incorrect assembly, meaning incomplete sealing or incomplete wetting. And any investigation of a filter failure or retesting should be described in the operating procedure. So what PDA goes about by saying is that if you have a failure of an integrity testing, it is not because of the damage of the filter only. There could be other reasons, because remember that you are assembling a filter on a housing. There is a wetting of the membrane which has to be done before the gas or the liquid passes through it. And in that scenario, if there is an incomplete wetting or there is an incorrect assembly, then also the integrity test could be failing, right? The method of doing an integrity testing is bubble point or your diffusion test or water intrusion test. In all these scenarios, you need a certain amount of wetting to be done for the filter integrity testing and to assemble the filter into that housing. In case of water intrusion, it is in situ, but other cases it is done in a special housing. So there could be a failure because of reasons of the testing itself, and this needs to be definitely looked into. To distinguish between the filter damage and possible test problems or artifacts, the following verification steps may be taken. This is again PDA technical report number 1026. First of all, that you have chosen the right integrity test method, the correct test parameters have been chosen, the correct wetting fluid and wetting procedures have been used, there are no leaks in the test system itself, the filter assembly temperature has remained stable and within specification during testing, equipment has been properly calibrated, test setup has properly been assembled and functions properly, and correct filter has been installed. All these factors play an important role during integrity testing. So we have to make sure that the right integrity test method has been chosen. Water intrusion test can be used for hydrophobic filters. You start to use it for hydrophilic filters, it will definitely fail. So you definitely cannot use a water intrusion test for a hydrophilic filter, for example. You will use a bubble point test, or you will use a diffusion test for it. The correct test parameters have been used, wetting fluid, this procedure. So remember, when we were talking about filter selection in the previous session, we said you have to take the procedure from the vendor. Why? Because they have done enough studies and will give you a procedure of how the integrity testing of their filter should be done. It may vary from one supplier to the other. So you take a Sartorius filter and look at the procedure and look at it as millipore, the same procedure might not be applicable. Very important to make sure that you've got the correct procedure for wetting the correct wetting fluid, and the wetting procedures have been used, correct test parameters have been used. The testing system, whether it is done in situ or whether it is done externally on the machine, there is no leakage in the system, because what are you trying to test? You're trying to see the pass through. You are at the end of the day, you are measuring a pressure drop. You are measuring what is the differential pressure on two sides of the membrane to determine whether your filter was integral or not. If there are other leaks in the system, this can also give you a false positive. And therefore, it is important to make sure that your test system itself is integral. The temperature remains stable. Why? Because with the change in temperature, the pore sizes may increase, the diffusion properties or the flow properties of the liquid will change, and therefore you need to make sure that the filter assembly remains stable. Equipment has been properly calibrated. Any equipment that you use, pressure gauges, differential pressure gauges, automatic pumping systems to allow the water to pass from one side to the other, to when you're doing water intrusion test, or having a proper air filter regulator to make sure that the pressure which is applied is correctly measured. All those instruments which are used have to be calibrated. The equipment itself has to be calibrated. It has been properly assembled and functions properly. There is no bug in the software. There is no issue in the software. So this is very important that you make sure that it is properly assembled and functions. And assembly is done with every filter. Functionality is a matter of proper preventive maintenance and care of the equipment. And of course, the correct filter has been installed. You are trying to, you sometimes get in a lab multiple filters, and if you mix them up and you give a wrong result because the data was completely different, and report a wrong result, you will have problems. You Because please remember that when a filter damage happens and you report a problem, you have to investigate the cause of it, and that till that investigation is completed and corrective action has been implemented, you may not be able to proceed with the same process of filtration that you have already painstakingly put together. So very, very important that any testing which is done taking into consideration these factors. Retesting protocol, the PDA technical report allows you to also do a retesting to confirm corrective action has been effective. Following retesting steps may be taken. Rewet the filter according to the specification and repeat the test. So if you find wetting was not done properly, you are allowed to do a repeat test by rewetting the filter. If it fails again, then you can apply more aggressive wetting condition by increasing the flush volume time, increasing the pressure, or applying the back pressure. If it fails yet again, perform the integrity testing in lower surface tension reference fluid to assess filter wettability changes independent of filter integrity. If the filter fails using the reference fluid, the filter fails the test. So this retesting protocol is something which has been introduced or has been recommended into the PDA technical report. While there is a mention of retesting permissible by the regulations, here it is very specific saying what is the sequence in which you should do retesting. You do a rewetting, you do aggressive wetting, and then you also do a surface tension test. If after all these three it fails, which means you have failed. So it allows you to still, because remember that when you manufacture a batch, the batch could be costing you millions of dollars. And if because of a false positive on a integrity failure of a filter, you have to discard the batch, you are blowing away millions of dollars of the company. Obviously, nobody's happy about it, but at the same time, you have to be sure that your filter integrity did fail. So by applying a retesting protocol, you are making sure that you are assured that yes, there was a failure before you take that decision to destroy $1 million batch. So this is very, very important. That's why the technical report is of great importance, because it gives you the cause, effect, and the methodologies of doing a failure analysis of integrity. Okay, so these are the failure analysis decision tree. I'm not going to spend time on it because we've talked about it just now in a brief summary. This is only putting it into tree, which helps in decision making process. Okay. Now, what are the key factors which affect the integrity testing? I have put in all the three tests here: diffusion test, bubble point intrusion. You will see that I have not put a pressure hold test here because of a simple reason that it is not widely used. So wetting liquid, test gas, membrane porosity, and system in terms of stabilization time, leak tightness, and temperature equilibrium are important factors when considering diffusion test. The same four factors are not applicable, are also there for the bubble point test, but the parameters are different. The surface tension of the wetting liquid, wetting angle, and temperature consistency and range become important for the bubble point test. The test gas is the applied differential pressure. At what increment are you applying the differential pressure becomes important, and the temperature consistency during the of the test gas becomes important, because remember the gas law, with the change of temperature, the gas expands or contracts. So it's very important to ensure that the temperature remains constant. Membrane should be completely wetted. Diameter of the largest pore should be known, and the system leak tightness is absolutely important in all the tests that we are talking about. In case of a water intrusion test, you should know what is the headspace that you are creating. You should be able to measure the headspace. You should be able to do wetting angle. Come on. Temperature consistency, again, what is the applied differential pressure and the temperature consistency of the gas itself. Contamination, whether it's a hydrophilic spot or a hydrophobic interaction. You could remember that if the filter was hydrophobic but had a chemical treatment to make it hydrophilic, you could have hydrophilic spots, or even in a standard hydrophilic membrane, you could have hydrophilic spots, and vice versa. On a hydrophilic membrane, you could have hydrophobic interactions. This normally occurs when the pleat and the cage in which this pleat is installed, at those interaction points such spots may occur. Porosity of the membrane is of course important everywhere, and then of course the stabilization time, thermal equilibrium become important. So these are the factors which will affect your integrity testing. Have to be cautious about making sure that these factors remain constant during the test. Okay, now, sorry, it jumped too much. Now, the technical report goes to saying what is a troubleshoot guide. So, if you don't see your test pressure build up, what could be the reason for it? Check all these things and make sure that it is not because of this before you conclude that there is a problem. Continuous venting of the unit, internal valve contamination and improper function, abortion of test during stabilization phase. So you are trying to stabilize the condition and suddenly there is an abortion of the test because it was insufficiently vented filter, or the filter was contaminated, or you installed the wrong filter. So these are all the troubleshoot guides which is given in this PDA technical report, Appendix D, Table D2. I'm not going to go through each and every point here for the paucity of time, but this is a very nice table which can be used as a reference to see that your integrity test, if there was a failure, what could be the reasons of those failure, and to investigate it and to take sufficient procedural and system controls to make sure that these conditions do not occur. Why is this table important? Whatever is in the light blue side of the table or the right hand side of the table, these are things that you have to take care in your protocol. You have to make sure that your protocol is written, your procedures are well defined, to make sure that none of these conditions are first of all existence when you're carrying out the integrity test. However, if you had a failure in the integrity test, then this guidance gives you a reason why no test pressure buildup might be happening, or measured volume may be wrong, or there is a failure of a bubble point or a diffusion flow, etc., and therefore investigate and allow you to retest, right? You can say I failed because there was an improper connection, there was an incorrect test gas, so because of this it failed. I'm resubjecting it to retest so that I can check whether the filter was really damaged. Okay. WIT troubleshooting. So what you saw here, you did not see a troubleshooting data on WIT. So this is again from the PDA technical report on why, what are the different factors which could lead to WIT pre-use and post-use failure. So the left-hand side is pre-use data points, right-hand side is the post-use data points. So you can use these indices, or you can use this flow chart and the data to define if your WIT failed or if your integrity test failed, why did it fail, and to make sure that you have taken enough measures that first of all these failures don't occur, and if these failures did occur, then how do you investigate it, and whether you can take a, with this, be able to take a decision on being allowed to do a retest of that filter. Okay, with this topic, I am done with the filter integrity testing. I'm going to jump to the next topic, or do you want me to stop here for any question answers? I am flexible on both. We have two more topics to cover, media fill and aseptic behaviors. So if you think that those three topics will become overwhelming and you may forget to ask me questions, I can give you a quick 5 minute time in which we can ask questions and we can move forward. So do you want to ask questions now on filter integrity testing? I will allow you to ask questions. Okay, if there are no questions, I'll move on to the next topic, media fills aseptic process simulation. Okay. Okay. Media fills are a very important part of sterile manufacturing process. It is nothing, it is also called as aseptic process simulation. So media fill is something that you would use as a terminology. You would hear that we are going to do media fills of a particular setup, or we are going to do a media fill of a new facility, or we are going to do a confirmation of the media fills. Um, but when you are searching for any literature, aseptic process simulation is the other terminology used for it. Throughout this presentation, both of these terms will be used as interchangeability. Media fills is something which is used very often. So when I'm talking, I'll use media fills more often, but aseptic process simulation and media fills is one and the same. Aseptic process simulation is also abbreviated as APS through this presentation. So aseptic process simulation is an essential activity for validation of an aseptic manufacturing process and is required by the regulators to demonstrate the aseptic capability of such a process. So what is this aseptic process simulation? It is a regulatory requirement where a documented procedure on an activity is followed by the manufacturer of the sterile drug product to ensure that the aseptic capability of the process is available. Okay, the successful program of media fill and aseptic manufacturing requires significant operator training, skills, and supervision, thorough maintenance, effective cleaning and disinfection, significant oversight of every aspect of the operation by quality assurance, and microbiological monitoring by quality control. So media fill is a program. It is just not a protocol that somebody in manufacturing follows. It requires the operators to be properly trained on aseptic behaviors, on aseptic processes, on the equipment, on the process itself. It requires the right skills. You cannot put probably an untrained or an unskilled worker there and expect him to perform this operation, because when you are talking about aseptic processes, you training and skill set becomes important. There is a certain amount of supervision which has to be done during routine operations, but also during media fills it is very important to carry out this supervision at all. The maintenance of the equipment has to be there thoroughly. Why the maintenance comes into picture? When you are doing the media fill at the initial stages, when you have just set up a facility, a new facility, then maintenance will not be a question mark, because you've just commissioned the equipment, they're good as new. They are new equipment, you are starting a new process. So when you do a media fill, you may not need to have done a maintenance. But maintenance does become important even in the initial stage, because the first operation, what are you doing? You are doing the qualification process. You run the equipment during OQ, during PQ. You may have taken some trial batches. You may have taken some engineering batches before you decide to do a media fill. In such a scenario, you have to make sure that during that period, after it was handed over by the vendor to you, whatever maintenance, routine maintenance has to be done is done. A lot of times people don't maintain equipments properly. The production is always under pressure of manufacturing. They have to keep on giving the product out, so the preventive maintenance schedules do not get followed in some industries. Of course, all the large manufacturers and now most of the companies recognize the importance of the preventive maintenance schedules and give proper time within the year to do a preventive maintenance of the equipment, because after all it's a machine and an equipment needs maintenance on a periodic basis, and you have to maintain the equipment. If you don't maintain it, it will fail. Effective cleaning and disinfection of the equipment or the whole process train is done. There is a sufficient oversight on every aspect of operation by quality assurance. So, and microbiological monitoring by quality control. What does this mean is that not only during media fills, but also when you have defined your procedures, your protocols, it is very important that multi-dimensional team, multi-departmental team from process side, from quality assurance, from quality control, from maintenance, from safety, look at the operations and the entire procedure to make sure that you are not going to compromise the asepticity or the equipment performance during the regular operation. What media fill is doing is just to make sure that you are simulating the aseptic process and doing the test so that you know that you are producing a sterile product repeatedly and consistently. So you do this media fill process simulation or aseptic process simulation or media fills on a periodic basis. You will do it initially at the time of a product. So whenever you start a facility and you may have a series of product, you will do media fills and then if you change the product or introduce a new product, you have to do media fills. Also, you will do media fills on a periodic basis that you may have evaluated the product. Now you're routinely producing it day in and day out. After 6 months, after 1 year, you have to repeat the media fill and make sure that your aseptic process simulations are there. Why? I will come to it as we go through what is done during media phase. An overall validation of aseptic processing is used to assess the contamination risk of an aseptic production process by simulating the manufacturing process using microbiological growth media instead of a drug solution. So what you use during media fill is not the product, but you will use a microbiological growth media and do the whole aseptic process so that the same way that you would run a formulation, filtration, and fill it into the vials, cartridges, ampoules, whatever, be your primary packaging material, you will simulate the whole process or run the process, but you will not use a drug solution. You will use a microbiological growth media. You will collect samples at different stages of unit operations, plate them to see whether your asepticity remained, your sterility assurance remained or not. This is a necessary part because sterility test used to release batch of sterile products has inherent limitations in detecting contaminated units in batch with low levels of microbial contamination. Due to limited number of samples that can be removed for destructive testing, this relationship has been evaluated statistically. So why do you need to do media fill is because in a regular routine batch, first of all, you are not going to test every vial for microbiology, right? You are going to only take certain samples because microbiological contamination testing is a destructive testing, which means if you had, say, 100,000 vials produced in a batch, you are going to probably take about 10, 15, or 100 vials out of that and subject it to microbiology testing. You cannot take all the 100,000 and test microbiologically. Can you? You cannot. So from that perspective, since you're using a statistical sampling methodology to do a microbiological test of a final product, and you are probably working in the detection limits could be low because you don't have a high growth media. You have a product which is there. It is not a high growth media for any microbiological contamination. So if because of the aseptic process you were introducing a microbiological contaminant, the it may not be detected on a batch because it is not the right media for the growth of that microorganism. But if you use a microbiological growth media and simulate the process, meaning the operator does the process, you do the connections the way you are doing it in the main batch, etc. If any contaminant gets introduced at any stage, the microbiological growth media will immediately be able to catch it because the it's the right growth condition. It has got the nutrients for allowing the microbiological growth to occur. Sterility assurance in aseptic processing requires contributing elements such as heating, ventilation, air conditioning, clean room environment, material transfer equipment, manufacturing process step, sterilization processes, sterilization filtration to be qualified and validated as applicable, and for the personnel to be trained and qualified. So you cannot form an aseptic process simulation unless your entire facility is qualified and validated, your manufacturing process steps are validated, your sterilization process, sterilization filtration processes are already qualified. So all the IQ, OQ, PQ that you did, all the facility validations that you do while setting up a facility or routinely maintaining a facility, the training of the operators, all this have to be already qualified before you say that I'm going to do a media fill. You cannot do a media fill when, let's say, half of the facility is not ready. You have to have the complete train from start to finish ready and should be in the actual production mode of with all systems qualified. Only then can you do a media fill. So remember this, very important, that media fill can be done only after everything is qualified. Simulation of aseptic manufacturing process using liquid microbiological growth media is required by regulators to demonstrate. So this is a regulatory requirement. And what does it involve? You cannot just prove it by doing one media run. You have to do 3 consecutive media runs in the specified clean room environment with the designated personnel, followed by repeat media simulation at 6 monthly intervals. So this is the regulatory requirement, that you have to do once in 6 months media fills, and you have to do 3 consecutive media simulations, and none of them should fail. Any media failure requires a thorough investigation and root cause analysis, and further media simulations may be required to complete the validation. So if you are doing a media fill or media aseptic process simulation, and if there is a failure, you have to first you have to go get to the root cause. You have to investigate it. Why did the failure occur? And only after you have satisfactorily answered that question and redone the media fill, 3 consecutive media fills, and they are all successful, can you continue to use the facility. So before you use the facility, your 3 media fills should have passed consecutively. Even when you repeat it once in 6 months, you should do 3 media fills and make sure that they pass. If there is a, let's say initially you passed, you started producing it, after 6 months when you were conducting it, media failure occurred. You have to investigate it, get to the root cause analysis. It may be because the operator needs a retraining either, or there was some problem with the equipment, there was a breakdown in an equipment, you change something. Whatever be the root cause analysis, that has to be done, investigation has to be done, it has to be rectified, and then the media fill has to be repeated, and only after you get 3 consecutive successful media runs can you get it to continue with the process. So very, very important that in aseptic processes, your procedures are well defined, that operators are well trained, your systems are well reviewed by everybody, so that you don't have this issue, because any failure of a media fill will set back your production capacities down by at least 1.5 months. My experience on media fill failure is that when you have to investigate it, it is a mammoth task, because there are multiple interventions that we are talking about, and in that investigation to get to the root cause becomes a very, very it's subjective. I may feel that it failed because the pipe was not okay, but if I have to get to the root cause, I have to have sufficient evidence that that was the reason for failure. It's like running a internal court case within your organization to investigate and find the root cause analysis, to have sufficient evidence proof that that was really the root cause analysis. Because if you do a wrong investigation and root cause analysis, you may take a corrective action, but what if the media fails again? Or okay, maybe you were lucky, it went through, after 6 months you again had a failure. You cannot afford to have media failures repeatedly. So obviously, media fill investigation is a thorough job. It needs the knowledge and sufficient experience for the people to do this investigation, and cooperation from all the people. Okay, very, very important. Okay, people have to tell truthful, don't hide facts. When investigation is happening, you have to come up and say, this is what I did, this is during the simulation. If some operation went wrong, and yes, there mistakes do happen, after all of us are human beings running the operation. During that, if any mistake has occurred, immediately report the mistake, because if there was a media failure or there was a product failure, then that becomes a reason for root cause analysis and investigation. Whatever corrective action has to be taken has to be taken. Okay, they are carried out in your standard conventional clean rooms, with grade A laminar flow. It could be with a grade B background environment. Filling environment may be further protected by wraps or isolators. Okay, this is just to tell you that the media fill is actually done in the actual facility. It is not done anywhere else. These are some of the regulatory expectations from a media fill, that it has to follow the current GMP and GMP annexes and guidances. There are various guidances. There is European Union and Pharmaceutical Inspection Conventions, so PICS and EU specifications. Then there is China GMP, there is US FDA, there is Schedule M, there's WHO, there is, if you are really exporting to Japan or Brazil, there would be further NVISA, Japanese. So all these guidelines have to be taken into account while defining your media fill strategy and to define your media fill simulations. Okay, you may also have references related to ISO, PDA documentation. So these give you a lot of insights into how to give your media fill strategies. So again, take into account the study of such documents. So I'm telling you that if you were in that position where you are expected to write down a media fill strategy, you need to know what all factors to consider. Okay. When you do a product submission while filing a investigational new drug application or a new drug application or a market authorization, the media fill report has to be filed to the regulator. So if you are filing for any of the new drug applications or INDs, NDAs or MAs, you need to give your media simulation documentation to the respective authorities, because they will review this before giving you the approvals. Okay, then you may need to support these media files such with other additional documents like training records, environmental monitoring reports, any deviations, routine deviations, any investigations that you may have performed, risk assessments that you may have performed. All these are documentation which go to support a new drug application and approval processes. The expectation of APS is twofold. It should achieve 3 consecutive media batches that meet the target acceptance criteria. The solution filtration process must be validated against microbial challenge with 107 colony forming units plus 12 centimeter of filter media using Brevidomonas diminuta, a small celled gram-negative bacterium suspended in a drug solution. So you saw this organism in the previous presentation. So here it is again coming back and telling you that your solution filtration process must be validated with this microbial challenge. Okay. Now, examples of media file run sizes and acceptance criteria for APS that have been incorporated in GMP Annex 6 and guidance include: when filling less than 5,000 units, 0 contaminated unit should be detected. A contaminated unit is considered cause for revalidation following an investigation. When filling 5,000 to 10,000 units, one contaminated unit should lead to an investigation, including considerations of a repeat media fill. Following investigations, 2 or more contaminated units are a cause for revalidation. When filling more than 10,000 units, one contaminated unit should lead to an investigation, or 2 or more contaminated units are cause for revalidation. So this is something which is given in the guidance, saying that when you are doing the growth media simulation, you are not going to fill 100,000 units, for example. Okay, so you may fill say only 5,000 units, you may fill 5,000 to 10,000, or you may fill more than 10,000 depending on the strategy. It also depends on the volume. There is a certain guidance which gives a relation that if your routine production size is 100,000, then you need to at least fill 10,000 or 1/10 of that size. If your routine production size is 40,000, then you need to fill, you cannot apply 1/10 formula, because nothing less than 5,000 units is permissible by the regulators. So very important. Now this is a criteria that if you are filling 5,000 units, none of the units should be contaminated, whether it's vial, cartridge, PFS, whatever it is. If there is even a single unit which is contaminated, then you have to do an investigation and then a revalidation. When you're filling between 5,000 to 10,000, If you find one contaminated unit, it leads to an investigation. Once you do an investigation and do a root cause analysis, you will do a repeat media fill, and after the investigation, if you get more than two units or two or more units which are contaminated, then you need to revalidate the whole media fill process. While doing the 10,000, one contaminated unit will lead to an investigation, two or more contaminated units are a cause for revalidation. So if you look at it, very strict, even one contaminated unit anywhere will mean that you have to investigate. So investigation has to be done even if a single vial fails or a single ampule fails, whatever is your primary packaging material. Contamination unit, in case of less than 5,000 unit, if you have a contaminated unit, you have to relook at your strategy, you have to relook at thorough investigation and revalidate the whole process. If you do 5 to 10,000, investigation is a must have, even if one was contaminated. And you may decide to repeat the media fill to see whether you are able to pass the media fill. So you, without leading to a revalidation of a process. And when you're doing more than 10,000, investigate the cause if you had one contaminated unit. But if you had two or more, then you have to go ahead with the revalidation. So very strict guidelines in terms of when you should do. Points to consider when designing a media fill study. Worst case challenge always comes into there. You have to include routine and non-routine investigation. What is routine and non-routine investigations? When you are running a standard process, you may be, let's say, assembling a powder charging container onto a vessel and then removing it. Or sometimes you just say, okay, I'll remove it at the end of the batch. Add a step saying that no, this is not a routine intervention, but let me remove that container, close the valve and see if something happens. So you, when you are designing it, you study the process very, very carefully and say, what are the routine interventions? Meaning intervention is nothing but where automated process is not running, a manual intervention happens or a sampling is happening, even if it is automated. You will, these are routine, which means that when you run the process, you are going to do these interventions on a regular basis when you run a standard process. Non-routine one, okay, I was taking two samples in a batch, one before filtration, one after filtration. Let me take two more samples. That's non-routine intervention. So you will design the media fill study based on routine and non-routine interventions. You will introduce some non-routine interventions through a proper scientific justification and say, I'm doing this because it is likely to occur. How do you determine this? You determine this by saying, no, no, while I will never do it because it is not required, then you don't just go ahead and introduce a non-routine intervention. But if you were likely to do it, okay, I have taken a sample, but that sample I found was contaminated or I took it in a wrong bottle. Happens, again, human error. In that situation, I want to take one more sample. That's a non-routine. It should not happen as a routine, but possible that you may require to draw an additional sample, maybe once in 10 batches or something like that, because of a reason that I just now said. In such a situation, you identify saying that this is why I'm introducing a non-written routine intervention here, because though I'm not going to do it during the process, sometimes I may need to do it. In a very rare case, I may need to do it. So let me introduce that and see whether my process is still remaining sterile or aseptic or not. If you have a lyophilization operation, you need to include it there. Microbiological growth medium, of course, you need to choose the right one. Unit handling, incubation, and inspection. So you need to make sure that all the monitoring is done. The incubators are right. So you incubate the samples, you draw the samples from different places and incubate them at the right temperature. And records and microorganism identification and operator training and qualification monitoring. Very, very important to make sure that all your operators are qualified, the entire facility itself is qualified, so all the records are checked, And if you find a microorganism, so you will do an investigation to see whether that microflora is present and characterize it. In any environment, you are in Hosur, you are in Jigni, you are in Hebagodi, you are in Bomasandra, you are in Tumkur, wherever you are, or you could be in Gujarat, you could be in Andhra Pradesh, you could be anywhere, you could be in US, Sweden, wherever. The microflora which is present in the environment is different in these different places. Even between 10 kilometer of a space difference, your microflora could change. So when a microflora changes, you should first of all know what is the microflora which is present. And when you see a microbial growth, yes, you are challenging it with B. diminuta, but you should be also be able to identify and characterize any other kind of a spore that you see in a growth media. What is that microorganism which got into your system? So that characterization is important. So when you are doing a media field study design, you will identify the microflora and say these are the different kind of microorganisms which could intervene and be present in my failures. Okay, apart from the challenge bacteria that you're using, but when you do an investigation, this data will help because you will then characterize or see that failure and see whether it was because of some of the microflora which was present. The microflora was not present at all in your environment. It was not present in the challenge bacteria. Then it becomes important to characterize that microorganism which you see in the growth media after a media fill failure, and to make sure that you find out the root cause for this. And that is which I what I will cover in the next session of aseptic behaviors, which is in the same presentation. So I'm not going to go through all these readable items. You can look at it, but again, how to define a worst case in terms of filling process, slowest filling speed, maximum fill volume, maximum batch filling conditions, maximum number of people in an aseptic area, do a shift change, operator breaks. Consider all these as a part of worst case challenges. Routine and non-routine, I spoke about. Lyophilization, if you have a lyophilization process, of course, the regulator says that you have to make sure that lyophilization process is also simulated, because while you may not actually do the lyophilization process because your bacteria is not going to get lyophilized, right? You may still need to simulate this in an possible simulation study so that you do not leave out any contamination which could be coming from the chamber or a non-sterile chamber. Okay. Media fill, growth media, what is the type of media you use? Different pharmacopoeias have a specification which is there. You can even prepare a media and show that it is supporting the thing. So anaerobic, aerobic microorganisms may be present, and accordingly you may want to use one or more media during the growth or the in the simulation study itself. So environmental monitoring during the APS, all routine and normal processes should be continued to maintain the clean room environment in qualified status. This includes particulate and microbiological environmental monitoring, which can demonstrate that the specified clean room environmental conditions are maintained. What does this mean is that when you are doing the media fill, you will maintain the clean room conditions as it is. During a clean room operation, you do non-viable particle counts on a routine basis, depending on the grade of clean room that you're operating it. And if you are working in a grade A zone, then there is a specified interval at which this has to be done. Both viable and non-viable monitoring of the environment has to be done. Apart from the parameters of pressure, differentials across rooms, the temperature inside the room, all these parameters which you would use during an actual process have to be maintained during media fill, have to be monitored, recorded. Okay, so very, very important that no aspect of your operation gets changed. You only introduce worst case scenarios into this, or more contamination into this, and make sure that you are still running an aseptic process. When I say contamination, through interventions and through more people, okay, not by deliberately introducing a contamination, then you will have a failure. Records, all the records of the APS batch have to be maintained, be it grade B, grade A conditions, whatever be your conditions, it has to be recorded properly. Record is the most important thing. The data integrity of this record is also very important. Okay. Operator training and qualification, very, very important, because ultimately people are operating a facility. How they use different aseptic, how they behave during an aseptic process, how they use different things. When you are using a process, if you are supposed to scoop something and charge into a vessel, that training has to be there. He has to do it in a certain way. All operators who do the operation have to be trained and qualified that they know how to do it. You cannot just, something spills, okay, nobody is looking around, there's no CCTV camera, let me pick it up and put it inside from the floor. Not allowed. So all these trainings are very, very important. During the media fill, you will have the quality people also standing next to you. You will have supervisors standing there to make sure that the people who are operating or performing the operations are well qualified. If they find that an operator is not qualified, that could become a cause of failure. But even if that was not failing, if they find that an operator was not behaving in the right manner or needed an additional training, they would record this and put them off the production shop floor and put them onto training. Very, very important that we do the operator training qualification properly. And what are the various training points are listed here: that sterile material should be handled only with sterile instruments. After initial gowning, sanitization has to be done properly. Personnel should not be directly in contact with the sterile product, container or components or critical surfaces. Rapid movements which create turbulence in critical environment should not be done. Movement should be slow and deliberate. Uh, Aseptic operator should not disrupt the lab or grade A conditions. So all these are important thing, and of course there is theoretical training, the kind of training that all of you are taking, understanding what is sterilization, how sterilization is done, what is the importance of sterilization, conditions, etc. Those kind of trainings have to be imparted to all the operators. Media fill has to be done by the operators themselves. Okay. Media failures, root cause determinations. You have multiple reasons why a media fail could fail. A key step in investigation is to identify the microorganism in the positive media file. So you need to characterize what is the microorganism which you detected during the media fill failure. Look at the environmental monitoring plates and see whether the same species of organism was available in those areas at the time that the media fill was performed. Okay, identification of species from colonies or plates exposed in lower grade adjacent to the clean room. So you not only expose the plates within the area which is subject to the media fill, but even the surrounding areas, to check that if there was any species which has some of the microorganisms which you may end up detecting. Okay, the review of the deviation should encompass preparation of and manufacturing processes. So the entire process, start to finish, from warehouse dispensing all the way to the product packaging, all operations should be covered, all HVAC parameters, all filtration parameters. So all the parameters of the process have to be recorded. It may also include review of CCTV records of the filling room in operation. So if CCTVs are present inside the rooms, inside the process areas, then you may want to review the CCTV records to just check what happened when a media failure occurred. Okay. In the investigation, different possibilities may provide evidence to support root cause determination, such as the ability to match the identification of an environmental isolate from current or recent batch with the identity of a contaminating organism of the failed media in the failed media units, or a significant processing discrepancy or error or an equipment failure. So if you dispatched a batch and it got rejected by the client saying that we have found a contamination, if you isolate the microorganism from such a return batch and identify that that organism is also seen when you have conducted the media fill, it becomes an important investigation point. So no uh, stone should be left unturned while doing an investigation. There are two figures which are available. This is something which I found in the ISP guidelines. So I've brought them here, the Ishikawa diagram and the potential root cause table. I'm not going to again get into detailed discussion here, but what I'm going to do is I'm going to leave you with this. So this is a media fill failure root cause investigation and identification using the Ishikawa diagram. So it talks about material, environment, operator, machine, and method, saying that what of these could have caused a failure. This is, you create an Ishikawa diagram or a fishbone structure diagram for your investigation that you carry out, and do a check on all these aspects, and then assign a cause to why the media failure occurred. This table here is going to give you the potential causes of media failures. I'm again not going to read through everything, but it talks about why an operator could cause to a failure and what is the mitigation plan against it. What happens when a machine failure occurs, environment failure occurs, method, even laboratory procedures have to be question, that did the person who took the sample plate it correctly? If that plating was an issue, then it could have led to a wrong result also. So false positives can sometimes occur because of laboratory procedures or interventions being done incorrectly, or samples being drawn incorrectly. Or failure to spot a non-integral vial, or a container or a vial had a crack and you did not inspect it properly, and that was allowed to go for the media fill, and that resulted in some ingress of the micro contamination leading to a failure possible. So all these stuff has to be investigated, and of course the material itself which is used, whether it's a packaging material, your depyrogenation process, your raw materials, damaged gowns and gloves. The maximum number of 483s have been there because the gloves have been damaged during use. They have had pinholes, and these pinholes have led to contamination being introduced into the sterile environments. Very important to investigate all this. Okay, typical media fill regulatory observations. So this is a table which gives you what are the observations which were made by the regulators during media fill, and this is a listing of it. Again, I'm not going to go through each one of them, but you see how important the media fill is, because the regulators deep dive into it, the your own people have to deep dive into it and get to the end root cause of failures. Okay, even the root cause analysis that you have done could be a wrong way of doing it. So regulators said your root cause analysis was defective, you did not do a root cause analysis properly, therefore you did not design the kappa properly, the corrective action was not defined properly, and therefore you had again a failure. So something which is very important when we talk about media fields. The last section of my presentation is around the aseptic processing behavior. People are the most important element in aseptic processing. Why? Because they are doing the processes. They are inside the room carrying out the various processes, pressing the buttons on the machines, charging reactors, charging formulation vessels, doing the formulation, monitoring the data as it is happening. So people have to have the right behavior and training inside an aseptic area. If they have the knowledge and follow good aseptic techniques, then you generally do not see failures. But if you do not have proper behavior, if you do not have appropriate controls or trainings, then you can introduce contamination into the process. Microbes are found everywhere, all around you and on you. So I have a statement which I make in when I am giving training on clean room. I say that the biggest contaminant in a clean room are always the human beings, because they are the largest source of microorganism. I was happy to see the slide flash up in one of the regulatory presentations that I was looking at. And I said, no, this is an important red dot which I see on the left hand side. So I took that from that slide and I said, this is a very important topic. People are the largest source of microorganism. Why? Why are we why are we murdering people? Why are we saying people are there? Because they have all these different microorganisms on there. They, when they come inside a clean room, from the skin you can see staphylococcus spp coming out. From skin you will see bacterium coming out. From feet bacillus and mold comes out. From skin other bacteria also comes out. You have streptococcus spp coming out from the throat. You have the E. coli in your gastrointestinal tract. All these microorganisms are already there. And if you look at it in terms of what is there, human sneeze, because a person inside a clean room can have sneeze. Uh, Normally they are talking, there is 1 million per milliliter of contamination in a human saliva, and human skin 2.5 million per 1 square inch of skin. This is the density of the population. Of course, we are not going to end up having human fecus in the clean room, but human skin, saliva, sneeze, all three possibilities there. Okay, so what can go wrong? If any gowning component needs a change or adjustment, the operator must exit. This is a good behavior. You should go and change it in the change room. If there is a hole in the glove, exit the room, plate and re-glove. Put another plate the glove which has failed, and then re-glove. If communication is needed due to safety or quality reasons, communicate away from the exposed product or component. So communication has to be imperative. You talk, sneeze or cough into the mask, return to the gowning room and change the mask. Because you have sneezed once, but you have coughed once into the mask, but that is now holding certain amount of microbial contamination. And it will go through if you are inside the clean room for more than 2-3 hours, because that contaminant has the right environment probably to grow. In an event of an emergency, when the operator needs to evacuate, then clean room gowning is changed upon re-entry, and shoe covers must be worn until dedicated shoes are disinfected. So if you have dedicated shoes, they need to be disinfected, and you must be protecting that with the shoe cover. If you evacuated a room for any emergency, sometimes people go out of the clean room for routine bio breaks or whatever it is. Generally, when once you enter 4 hours, you are not allowed to go out, but that's the training. But in case you need to go for any reason, yes, nobody will prevent you. But when you re-enter, the gown has to be complete. You cannot wear the same gown that you wore earlier. That is discarded. You go out. So generally, in clean room designs, a lot of good facilities, they have an exit path which is different, where you discard your gown and you cannot access that same gown which was worn from the other side. Sometimes when you have the same change room for exit and entry, this possibility may occur. So it is not right or wrong. This is a training and practice which has to be followed. Gloved hands contacted any part of gowning apparel, then it has to be sanitized with alcohol, and that disinfectant has to be changed. Okay, remember that we are a walking culture. We have microbial contamination all over our body, into skin, clothes, shoes, onto our garments. Everywhere there is a microbial contamination. Now, therefore, also when you wear street garments and go into a facility, generally in sterile manufacturing, you are not allowed to take that street gown into clean rooms. You will be given a change room where you remove your street garments completely, and you are given clean garments which you wear, and then you overgown on top of it as you go into respective higher grades of clean rooms. Okay, so this is some data on what is the micro carrying capacity per minute on these kind of garments, and what is the average particle shedding of greater than 0.5 micron which can be shed per minute. Okay, so very, very important to understand. This is the gowning protocol that you follow in different grades. Again, for paucity of time, I'm not going through each one of this. You can read up this. Okay, what is important, that there should be sufficient appropriate personnel suitably qualified, trained and experienced in manufacturing testing or sterile product. So even if, let's say, you called me to your facility, and you wanted me to enter into your sterile areas, you have to give me the training of your procedures and protocols before I'm allowed to enter with you. Okay, even though you may be accompanying me, you have to impart me a training irrespective of what my previous training may be, because I'm entering a facility which has got defined procedures and protocols for entry into clean rooms. I cannot be allowed to enter just because I have experience of 30 years. No, you have to take me through the procedure of what I need to follow as aseptic practices before I enter into your area. So generally, if you call any consultants or if you call even auditors, you are supposed to give them a copy of the procedures to be followed in aseptic areas before they are allowed to enter those areas. Okay, and they are generally accompanied by a person from the factory so that they can observe that this person is not making a mistake. So very, very important to make sure that it is not only applicable for people who are working, but even for visitors who are coming in, there is a certain basic training to be imparted before they are allowed to enter aseptic areas, if you are allowing them to enter aseptic areas. Generally, visitors are not allowed to enter aseptic areas. You create visitor galleries or visitor corridors, they which go around, which require minimalistic gowning of a grade D, or even routine gowning with just a overcoat, a head cap, and a shoe cover, and you allow them to walk. They see through windows all the facilities, but auditors will enter, quality people will enter. So even if a new quality control person, he's got 20 years of experience, joins your facility, if he is not trained in your procedures, he cannot be allowed to enter your aseptic area. This is very important, and the reason I'm saying this is that I don't want to murder just the the operators and say operators are not trained. The people who are operators are not trained, or the supervisors not trained. Even with 30 years of experience, if I came to your facility from another organization, I need to be trained to specific procedures of your facility. Very important. Okay. So again, around the same thing of hygiene and cleanliness, health is very important. So people who operate inside the facility should be healthy. If they have any disease, even if they have a slight cough, fever, any of those symptoms, they should first of all voluntarily report it, and because they can potentially bring in microbial hazard into the facility. So they should voluntarily give it as an information, and the assessment is done by the safety officers to say whether they are fit to be allowed to continue to work in a clean room, or they should be removed from that and somebody else should be put in there. Okay. So health condition is very important because you can bring in other contaminants which are not routine contaminants which are there, or standard microflora which is present. Okay. You need to have identified clean rooms. You cannot sometimes reuse the same garments unless they are clean, sterilized. In some facilities you will see single use garments, some facilities recycle garments through proper procedures. But whatever it is, you are supposed to make sure that you have appropriate gowning procedures defined through a standard operating procedures. Okay, gloves should be regularly sanitized. Mask and gloves should be changed at least every working session. So if you have a working session for 3 hours before you go out and come back, or you go out into another room, every time you enter a new area, please make sure you change your gloves. You sanitize the gloves. Your goggles are sanitized. Your wristwatch, cosmetic jewelry, nothing is allowed. Please don't ask it to be taken. You are, irrespective of your, if for your beliefs of faith, you have to remove all the wristwatch, cosmetics, jewelry into the clean areas. You cannot take any of that inside. So, very, very important, because even a small contaminant can create a problem for the patient. Okay. Compliance to aseptic gowning procedures is very, very important. Again, read through the gowning procedures. What is important? Be trained and qualified, be medically qualified, follow good aseptic behaviors in a consistent manner. With this, I end my session. Thank you for paying attention. I will take questions. I know we have exceeded the time, but it does not matter. This was an important session. So I'll take questions if there are any on any of the three topics that we covered: filter integrity testing, aseptic behaviors, media fill. Please shoot your questions, ask me questions, and then probably I'll spend a couple of minutes before I sign off. So let's open the session for question and answers.
Sir,
Yeah.
what is the importance of the wetting procedure, sir, of the membrane? For example, in the bubble point test and in the diffusion test.
Okay. Let me go to the diagram there. Look, what is the bubble test itself? Is that you are wetting the membrane, then you are pushing a compressed air, and the wetting of the membrane creates a surface tension. As you increase the pressure, it overcomes the surface tension, all right, the force of surface tension, and opens the pore up. If you don't wet it sufficiently well, then what can happen is that an unwetted surface may be available through which the compressed air will easily pass. Any gas or liquid will start to flow through the shortest path what is possible. If you don't wet the membrane properly, when you are doing a bubble point test or a diffusion test, if it is not wetted properly, this gas will bypass the water layer or the wetted surface and will go through the non-wetted surface easily out and give you a wrong result. So before, even at this pressure, you will suddenly see a bulk flow happen. So wetting is important, because if wetting is not proper and the surface tension is not proper, you will get a wrong result of a bulk flow. Same thing for diffusion, because in diffusion also what are you doing is that you are holding the water and allowing the gas to pass. If there is no wetting is inappropriate, this gas will start to diffuse out easily through another non-wetted area and give you a wrong result. Understood?
Yes, sir. Thank you, sir.
Okay. Any other questions? Okay. So if there are no questions, I'm just going to take 2 more minutes of yours, because this is my last training session with this batch. You will of course have the quiz questions which will come through Dr. Vasant and the MS and the team Biocon Academy. which will be shared with you, and you will receive those questions on Friday by Friday, and you have to review those. So first and foremost, I would like to start by saying a big thank you for this group of students who have the trainees who have been participating in this, have been interacting. Though I have a complaint that there were not enough questions asked per session, but that's fine if you have known the principles, or if my presentation was sufficiently elaborate to give you the right training. I'm happy about it. So thank you, a big thank you for that. I would also want to seek a feedback. I will reach out to Dr. Vasan, Dr. Vishal, and Biocon Academy. I would like this trainees to tell me or anonymously to give me a feedback on the training sessions that I have held. What did you like about the training? What you felt could have been better? What did you find missing? What more information would you like to have seen on each of the topics which was presented to you? You have all these presentations on the Moodle portal. You can review them and give a honest feedback. It's not for judging your capabilities, but more importantly for me to improve the training that I impart to students like you, to trainees like you. So in the future, when I see Gen Z, as they call you, right? I don't know what they call now the generation, but I think it's called Gen Z. When I'm training, where you have access to a lot of information through internet, through other technology advancements, from the experience that we have from a work that we have done over the last 30 years of work experience, how can we better this training modules so that the students are well prepared when they go out there in the industry. So I would seek that feedback to Dr. Vasan, Dr. Vishal, but I want each one of you to provide honest and fearlessly, please be very honest, brutally honest, and give a proper feedback so that it helps me improve my sessions in the future as a trainer. It's a test of the teacher, as they say, right? And I would like to go through that honest review from you. And yes, you can reach out to me as students at any given time as training in your life. My email IDs would be available. So even in when you start your work career after you have left this course and got into in the industry, you're most welcome to approach me from a perspective of sending me questions if you had doubts, and I will try my level best to give you the response to those questions. And I wish you all students the very best in your future, success in your careers as you grow into this industry, pharmaceutical industry, ever growing, challenging industry, because the regulations keep on evolving. New insights come into picture, new technologies come into picture, and as a result, it's a dynamic area of learning. And I loved doing these presentations and training sessions because at some of the topics which were presented, though I did not have a depth of the knowledge when I did the training sessions, it gave me an opportunity to read through more about it and then come prepared so that I could give these training sessions. So thank you and all the very best. And I would like to, not last but least, thank the Ramaya Institute, Sinjin, and Biocon Academy for providing me this opportunity to be your trainer and present these topics.
Sir,
Thank you very much,
sir,
students.
1 minute, sir. Sir,
Yes, Irene.
sir, so I would like to take this moment for express the immense gratitude for your classes and your guidance.
Thank you.
As we all know, you are a very busy person, and in this busy schedule, you have given such a great dedication to create these PPTs and to give these classes. And it was very knowledgeable, very informative, and very useful, sir. Sir, your dedication to teachers was really admirable and always thankful for you, sir. Thank you once again.
Thank you so much, Irene, for those kind words. I hope that I have made some difference to your learning stories and your learning path, and I would like to continue to contribute to that. Thank you, Dr. Vishal, for moderating these sessions. Also, Dr. Vidya and Dr. Vasant in some of the sessions, earlier sessions were coordinated by Dr. Vidya. So thank you very much, and I hope to see you in the industry, maybe come across to you as employees in some organization where I would go and work, or even opportunities which may be available within the firms that I deal with. So, all the very best to all of you. Have a very great week ahead, and further learning as you continue through this batch of Biocon Academy sessions. Wish you all the very best in your careers.
Thank you, sir.
Thank
Thank
you,
you,
sir.
sir.
Thank you, sir.
Take
Thank
care
you,
and
sir.
bye bye. See you.